Assalamu alaikum wa rahmatullah, everyone. I hope you're all having an incredible day. My name is Mohamad Behairy. I am a third year medical student. Inshallah, today I will be giving you the PAL about all the lectures post-midterm in MOL 2. I want to clarify something. This isn't simply a practice question solving session. This isn't simply a review session, not even a high yield session. This is every single thing you're ever going to need for the final exam, inshallah. I made sure that this PAL session will basically be 13 PAL sessions in one. Yani all the lectures, every lecture will take its fair share. I will explain to you everything that you will need to know. Yani wallah, even if you didn't even open your MOL slides yet, inshallah, this PAL session will be more than enough to ace the final, inshallah. All I need from you guys is to listen to me, take my words seriously because everything I'm going to explain is important. Oh, come on, review this slide and this PowerPoint, sorry, to the best of your ability because every single thing in this condensed PowerPoint is important. Before I start, I just don't want you to be scared because if you notice, you might see that this PowerPoint is 161 slides, but in reality, 70 of them are practice questions, 13 are checkpoints, 13 are the title slides. But all of the lectures of post-midterm Mol 2 have been explained, inshallah, in all these 60 slides. And yes, they cover everything you need to know. خلاص? But without further ado, let's start with وتطمنوا. You guys, inshallah, are in good hands. We're going to start with the first lecture, which is fatty acid synthesis. All right? We all know fatty acids are basically molecules in the body, and we simply want to know how we create them in the first place. All right? Here we have the mitochondria, and here we have the cytoplasm. In the mitochondria, we have citrate. It's always there in the mitochondria, and you guys probably remember it from the citric acid cycle. So citrate is actually a mix of acetyl-CoA and oxaloacetate. Now, why is citrate important here? It only has one function, by the way, because what we're going to use for the synthesis of fatty acids is acetyl-CoA. But there is one single issue. Acetyl-CoA has to go to the cytoplasm in order for it to synthesize fatty acids. But acetyl-CoA can't just enter the cytoplasm and exit the mitochondria on its own. It has to be transported through something. This something is citrate. So acetyl-CoA and oxaloacetate will become citrate, and this citrate will leave the cytoplasm and break down again into acetyl-CoA and oxaloacetate. يعني للأسف, it's a complex process. In an ideal world, acetyl-CoA would just leave on its own to the cytoplasm, but it can't do that. It has to be in the form of citrate, and citrate is the molecule that can freely exit and leave the mitochondria, and then this citrate will divide once again into acetyl-CoA and oxaloacetate. And this is actually the first step of fatty acid synthesis. I want you guys to remember that this breakdown in the cytoplasm happens through the enzyme ATP citrate lyase. And this enzyme will break down citrate into acetyl-CoA and oxaloacetate. Now, خلاص, we have what we want, which is acetyl-CoA, and everything from here on will depend on this acetyl-CoA to end up with a fatty acid. This acetyl-CoA will undergo a change. It will become something called malonyl-CoA. And how does this reaction happen? Through the enzyme, which is very important, more important than all of our lives. Acetyl-CoA carboxylase. I want you to remember this enzyme very well. It is the limiting and regulated enzyme of fatty acid synthesis. One other important thing I want you to know. This enzyme can only work when there is biotin. So if you get a question saying there is a biotin deficiency, you will know that acetyl-CoA carboxylase is affected. طيب. What will happen to this malonyl-CoA? It will become palmitate. Palmitate is the fatty acid, all right? The fatty acid that we synthesize in our body is called palmitate. And malonyl-CoA to palmitate happens through the enzyme fatty acid synthase. طبعا, it's more complex than simply getting malonyl-CoA to palmitate, but we will go into detail about that later on. For now, I just want you to take a look at this, appreciate the beauty of this because this is everything about fatty acid synthesis. تمام? So three important enzymes: ATP citrate lyase, acetyl-CoA carboxylase, which needs biotin, and fatty acid synthase. طيب. Something important I want you to know about fatty acids. We were just talking about how our body creates fatty acids, but what I want you to know is that not all fatty acids can be created in the body, especially linoleic and linolenic acids. These are fatty acids that can't be created in the body. We can't create them ourselves. We have to get them from food, outside sources. طيب, you might get a question in the exam. Why can't our body synthesize those two fatty acids? It's simple because we lack a certain enzyme. This enzyme is called desaturase. Desaturase is an enzyme that adds double bonds, and these double bonds will give us these special fatty acids. إحنا humans, we don't have desaturase, meaning that we cannot add these double bonds, which would lead to us failing to create linoleic and linolenic acids. So just keep that in mind. Something else I want you to know is that linoleic acid and linolenic acid are very important. Why? Because they give us eicosanoids, which are hormones that give us prostaglandins. But let's say someone does not take fatty acids from outside sources. They will have deficiencies in prostaglandins and eicosanoids. Prostaglandins are very important inflammatory mediators. They are very important molecules, and that's why fatty acids must be obtained by a person de novo and the ones that we can't get ourselves from the outside. Right, so fatty acid synthesis. We said that the synthesis of fatty acids occurs in the cytoplasm, where of liver and adipose tissue, cytoplasm of the liver and the adipose tissue. And we said that the process will use acetyl-CoA that first is transported from the mitochondria to the cytoplasm by what? Citrate. But how does citrate do that? How does citrate go out to the cytoplasm? By something we call malic enzyme. So malic enzyme will take the citrate out. As we said, citrate that is in the cytoplasm will then be divided into acetyl-CoA and oxaloacetate. How? By ATP citrate lyase. Then, acetyl-CoA carboxylase, which again, the rate-limiting enzyme, the most important enzyme that will need biotin alongside carbon dioxide and ATP, will give us malonyl-CoA from acetyl-CoA. And last but not least, malonyl-CoA will turn into palmitate by fatty acid synthase. But I want to talk a bit more about acetyl-CoA carboxylase because I want to drill this enzyme in your heads. Again, it is the regulated, rate-limiting, and committed step. Takes acetyl-CoA and changes it into malonyl-CoA. And it has three functional parts. You don't have to memorize them in detail, just know biotin, biotin. Biotin is everything for this enzyme. Now, regarding this enzyme, I want you guys to know what's going to activate it and what's going to inhibit it. But before we get into that, I want you guys to understand why we synthesize fatty acids in the first place, because wallah, that will help you understand a lot. طيب, when we have acetyl-CoA and all of this energy in the body, we have to store it. The body stores energy. If we can't use it now, if we don't need to use it, then we have to store it. صح? But energy can be stored as fatty acids. However, when the body doesn't have energy, it needs energy, it's hungry, it's starving, that's when it will break down the fatty acids, which is opposite of synthesis, oxidation. It will break down the fatty acids to give us this energy. So because of what I just said, I think you will be able to deduce that when there is insulin, meaning we just ate, we are full, we have a lot of sugar to the point where insulin is being secreted, it will activate acetyl-CoA carboxylase. Why? Because we want to turn this extra energy and sugar into fatty acids, which will be stored. However, when we have glucagon, meaning we are hungry, we need sugar, glucagon is being secreted, in that case, we will inhibit acetyl-CoA carboxylase because we don't have energy to make fatty acids. We want to break down the fatty acids, if anything, to get the energy from them. طيب, how does insulin activate acetyl-CoA carboxylase? By dephosphorylation. When you remove the phosphate, you dephosphorylate the enzyme, it will be activated. Glucagon, on the other hand, through AMPK, important, remember it, it will add a phosphate. It will phosphorylate acetyl-CoA carboxylase, leading to it being turned off. Now, this should be very straightforward and easy. Citrate will obviously activate the enzyme. Why? Let's go back. Because citrate is basically the first step of this reaction. But when we have a lot of the citrate, the reaction will go downstream. So acetyl-CoA carboxylase will be activated. However, when we have a lot of palmitate, it will induce a negative feedback to acetyl-CoA carboxylase to stop, and therefore it will inhibit it. طيب, what else do we have? Malonyl-CoA. Guys, this is very important. I want you to remember it because it will come in the upcoming lectures. Malonyl-CoA, which is this one. What does malonyl-CoA do? One might assume that it will inhibit acetyl-CoA carboxylase. ليش؟ لأنه it's the product of this enzyme. But no, malonyl-CoA, in fact, doesn't directly interfere with fatty acid synthesis. However, it interferes with fatty acid oxidation, the breakdown of fatty acids. And what does malonyl-CoA do? It inhibits an important enzyme in fatty acid oxidation that is called CPT1, the carnitine transporter. We're going to come to this enzyme later on. You will know it, and we will mention malonyl-CoA again. But now I want you to just remember this, that malonyl-CoA indirectly activates acetyl-CoA carboxylase. How? By inhibiting fatty acid oxidation through the inhibition of CPT1. Tamam? Then we have epinephrine. Epinephrine, guys, is like glucagon. You're hungry, you're tired, you're stressed. So this isn't the time to create storage fatty acids. This is the time to break them down for energy. So a high carbohydrate diet would give us high energy, which means we want to store them. We want to store this energy, so it will activate ACC. However, a high fat diet or fasting, meaning we don't have a lot of energy, would lead to the inhibition of this enzyme. Now, guys, we're going to move into fatty acid synthase. Do you remember when I told you guys, معلش, I'm going back every here and there because I want you guys to understand very well. Do you guys remember how we said that malonyl-CoA will become palmitate through fatty acid synthase? I also told you guys that this reaction is simpler than just malonyl-CoA palmitate. We're going to zoom in at this section, malonyl-CoA to palmitate, and it will take us to this slide. What does fatty acid synthase does? What does it do? It does four different things, all right? And I want you to memorize them. Condensation, reduction, dehydration, and once again, reduction. By the way, in fatty acid oxidation, which is the opposite of synthesis, we're going to have these same four, but relax. So reduction becomes oxidation, وهكذا. But let's leave this to its time. So I want you to remember, condensation will give us what? It will give us a beta keto product. They might ask you in the exam, which reaction in fatty acid synthase cycle will give you a keto product? You're going to be like condensation. The first reduction will give you an alcohol. Dehydration, obviously, will give you water. This isn't very important. And the second reduction will give you the saturated acid chain, which is the final product. This cycle will repeat almost 7 times until we can have 16 carbons, and that's when we have palmitate. Tamam? So please memorize condensation, reduction, dehydration, then reduction. So fatty acid synthase. Now, something important I want to note about fatty acid synthase. Remember how we said in acetyl-CoA carboxylates needs biotin? Fatty acid synthase needs NADPH. خلاص؟ احفظوها. It needs it or it won't work. So fatty acid synthase, I want you to know something, that fatty acid synthase does the reactions in something called an acyl carrier protein. You don't need to know what this is. Just know in all these reactions, the condensation or blah, 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 they happen on the acyl carrier protein, okay? This is for completion's sake. An acetyl transcyclase and malonyl transcyclase are two molecules that will take the substrates and put them on the acetyl carrier protein so that fatty acid synthase can work. As we said, 4 steps. Condensation gives us the beta keto acid. Reduction 1 will give us alcohol. Dehydration gives us the double bond and water. And reduction 2 will give us the saturated acyl chain. Nothing new. As I said, fatty acid synthase requires what? NADPH. Very important. طيب, what are the sources of the NADPH? Yani, how can the fatty acid synthase get NADPH? From 2 things, either the pentose phosphate pathway or the malic enzyme. Do you guys remember what the malic enzyme does? We mentioned it very early on. Sorry, I'm going back, but wallah, I just want you to understand everything. But we said it here. Malic enzyme is what takes citrate from the mitochondria to the cytoplasm in the very first step of fatty acid synthesis. Well, this malic enzyme will also give us a NADPH that's needed for fatty acid synthase. Now, last but not least, I want you guys to know إنه elongase من اسمها elongate. It's an enzyme that will add more to the 16 carbons. يعني palmitate is 16 carbons. It's already so big. If we want to make it longer, we elongate it by elongase. Desaturase, we already talked about this enzyme, how humans lack it, but it desaturates fatty acids or adds double bonds. تمام? طيب, last part of this lecture, it's very important. After we get these fatty acids and we synthesize them, وين نحطهم? The answer is triacylglycerol. Because all of these fatty acids, we said we're going to store them. صح? They are storing energy. We have to keep them somewhere. We keep them in triacylglycerol. Triacylglycerol is basically three fatty acids and glycerol 3-phosphate. We're going to talk about glycerol, but just remember, in tag basically will contain the fatty acids. You don't need to know anything here. All I want you to know is that tag has three fatty acids and glycerol. طيب, how do we make glycerol, the glycerol 3-phosphate? Okay. It's in the glucogenesis pathway, if you guys remember it. But all I want you to know is that glycerol 3-phosphate comes from pyruvate, the pyruvate oxaloacetate pathway. And an important enzyme that is associated with forming glycerol is PEP, carboxykinase. Just remember that this enzyme is associated with glycerol. Tamam? And with that, the first lecture is done. We're going to go through practice questions. I won't go into detail a lot. I want you guys to just pause and answer. I'm going to read it very quickly. حتى I might not read it. Just explain it and tell you the answer. تمام? So, for this question, it's asking you what molecule transports acetyl-CoA to the cytoplasm? We already said it's citrate. Second question, I'll give you the chance to pause it. This woman basically has a fat-free diet. What deficiency could she have? She doesn't get a lot of fatty acids. What molecule depends on the fatty acids that come from outside? We said prostaglandins through eicosanoids, which are linoleic acid and linolenic acid. Next question. They're talking about the reason for the special requirement in loop. Why can humans not do linoleic and linolenic acid? This is another name for these acids. إحنا humans, we lack desaturases that act beyond the C9 position for double bonds. We already said that. Next question. Talking about a female with fatigue, anorexia, and she appears very malnourished, and we suspect a what? Biotin deficiency. Yes, you see biotin deficiency, what conversion will be impaired? أول شيء, the enzyme that's impaired is acetyl-CoA carboxylase, صح? طيب, what conversion will it impact? Acetyl-CoA to malonyl-CoA. Last question, I believe. Insulin and glucagon both take part in regulating acetyl-CoA carboxylase, so which sentence is correct? It will be this one. Insulin will activate the phosphatase to remove phosphate group from acetyl-CoA carboxylase. Why? Insulin means we are well fed, we have a lot of energy, but we want to change this energy to fatty acids to store them. Glucagon, it doesn't activate phosphates. It activates AMPK, which phosphorylates the ACC, leading to its deactivation. So that's the first lecture done. We're going to go to fatty acids oxidation. Again, guys, wallah, يعني everything I'm saying is going to be everything you need, inshallah, for the final. Now, this lecture will be easy. Why? Because it's very similar to fatty acid synthesis. But now, حنمشي بالعكس. 2 slides ago, what did I tell you guys was the fate of fatty acids? Where are they stored? I told you they are stored in triacylglycerols, the tags. So أكيد, fatty acids breakdown oxidation will start with tag. Tag, we said, it has fatty acids and glycerol. طيب, but what enzyme will break down this tag to fatty acid and glycerol? It's a very important enzyme, and I want you to remember what activates it. The enzyme is lipoprotein lipase. It's a lipase, breaks down. And it's activated by something called apoprotein C2. We will talk about apoprotein C2 in the lipoproteins lecture, but for now I want you to remember tag is broken down to fatty acids and glycerol by lipoprotein lipase, which is activated by apoprotein C2. طيب, something I want you to know. Fatty acids, where are they synthesized? We said cytoplasm. Where are they oxidized, broken down? All the way in the mitochondria. But now we're not even in the liver. We are in the plasma. We are somewhere else. We need to get to the cytoplasm of the liver, then the mitochondria of the liver. Right, first step is to go to the cytoplasm. How can the fatty acids go to the cytoplasm? They use albumin. They bind to albumin, and through albumin, they will go to the cytoplasm. تمام? So خلاص, fatty acids successfully made it to the cytoplasm. But can it just go inside the mitochondria? No, it has to be activated. So fatty acids will be activated. What is this? What happens in this activation? We have ATP-dependent acylation of coenzyme A. You don't need to know the details of this. Just know that there is acylation of coenzyme A, and this causes fatty acids to be activated. طيب, what is the enzyme that does this acylation that will result in the activation of fatty acids? Very important enzyme. We're going to talk about a disease related to that enzyme, okay? So the enzyme is long-chain fatty acyl-CoA synthetase, LCAS for short. Remember this enzyme. It's the enzyme that's responsible for activating fatty acid. Long-chain fatty acyl-CoA synthetase. تمام؟ طيب خلاص now alhamdulillah we have the activated fatty acids. But I'm gonna give you now an example, a real life example. It doesn't happen, but hypothetically speaking, we're gonna pretend that it happens. Without how you're gonna understand what's gonna happen next. تمام? Let's say cytoplasm is Saudi Arabia, mitochondria is the United States of America. I am the activated free fatty acids. Hello, the activated fatty acids. I want to go to the United States of America, which is the mitochondria. In the visa passport control in the airport في America, they tell me, okay, listen, you can enter the airport, but you need to enter with an American citizen. And this is exactly what happens to fatty acids. Fatty acids will try to enter not inside the mitochondrial matrix, but the inter mitochondrial membrane, which is basically like the airport before you actually get into the country. How does it enter? Through CPT1. Do you guys remember CPT1? I'm going to go back. I want to remind you. We said there is something that inhibits CPT1. Malonyl-CoA. It inhibits CPT1, so it inhibits fatty acid oxidation. So malonyl-CoA supports fatty acid synthesis. Now we are talking about the CPT1. I told you guys it's going to be important. So CPT1, C is for carnitine. It's a carnitine transporter. And this is the rate-limiting enzyme, by the way. CPT1 will tell the activated fatty acid, listen, you can go inside the mitochondrial membrane, the airport, but you have to take carnitine with you. ما هو, it's a carnitine transporter. We are going into a carnitine-dominated place. So it's like, okay, you can go inside, but take carnitine with you. But the activated fatty acids will take carnitine. So this is the function of CPT1. It's the rate-limiting enzyme. It gives carnitine to the fatty acid so it can go inside the mitochondrial membrane. After that, خلاص. After you go through the airport, the passport, visa control, وما أعرف إيش, CPT2, which is the last thing in the airports, basically, it's going to be like, خلاص, you successfully completed the security checks. You don't need the carnitine anymore. You can enter the mitochondrial matrix, which is the country, خلاص, alone, without the carnitine. This is the activated fatty acid, AFA. But what is the function of CPT2? CPT2 will remove the carnitine from the activated fatty acid, will let it go inside the mitochondrial matrix. Well, guys, there are deficiencies in CPT1. We're going to talk about what they lead to. Deficiencies in CPT2, وكمان deficiencies in carnitine. But for now, what I want you to remember is that CPT1 is the rate-limiting enzyme. What will it do? Give carnitine to the activated fatty acid so it can go inside the intermitochondrial membrane. Then CPT2 will take the carnitine back and let the activated fatty acid go inside the mitochondrial matrix. بعدين what will happen? خلاص, beta oxidation. The thing we've all been waiting for will happen. And what will beta oxidation give us? I want you to answer this yourself. Before I show you the answer, we're going to go back to fatty acid synthesis. What is the most important thing in fatty acid synthesis? The acetyl-CoA. Who will give us everything? So in fatty acid oxidation, what is going to be the end product? Acetyl-CoA. As simple as that. تمام? Or just a side note, oxidation of long-chain fatty acids, it will happen in the peroxisomes. It will happen where? In the peroxisomes, not in the mitochondria, or by another enzyme called catalase. Now we are talking about normal fatty acids, how they're broken down in the mitochondria through the process I talked to you about. However, if we have a very long chain fatty acids, it will be broken down by catalase in a peroxisome. Tamam? I hope this diagram is clear. طيب, so activation of fatty acids. First of all, we want to know in the triacid glycerides, tags, are hydrolyzed into fatty acids and glycerol. How? Through the enzyme lipoprotein lipase, which is activated by apoprotein C2. We already talked about this. So the free fatty acids then go to the cytoplasm. How? By albumin, the cytoplasm of the liver. وبعدين it will make it to the cytoplasm. Fatty acids are then activated. احنا قلنا before you enter the mitochondria, you have to be activated. How are they activated? By long-chain fatty acid CoA synthetase, which is LCAS. I told you guys there is a disorder associated with this. It's called adrenoleukodystrophy. I don't think it's very high yield, but again, I told you that this file session has everything you need. So if you see something in my slides, you memorize it. Adrenoleukodystrophy is when we have a deficiency in LCAS, which means that what will happen? We can't activate the fatty acids, so there will be buildup of these long-chain fatty acids. They will build up. They can't be activated, or if they can't be activated, they can't be broken down. So they're gonna build up. Right, let's move on to CPT1 and CPT2. I already told you they're very important. We explained their functions. Let's go through them again. We said that CPT1 is located on the outer mitochondrial membrane and it transfers a carnitine to the fatty acids. And CPT1, once again, it is the primary, rate-limiting, regulated step of fatty acid oxidation. Once again, I'm going to repeat the thing I've been saying 70 million times. It is inhibited by malonyl-CoA. We said it in the first lecture, if you guys remember. طيب, how does malonyl-CoA inhibit CPT1? By insulin. Well, this has been asked before. Insulin will push malonyl-CoA towards CPT1 to inhibit it. تمام? طيب, obviously this would prevent the body from breaking down fatty acids at the same time as trying to synthesize them. So why does malonyl-CoA أصلا inhibit fatty acid oxidation? Because when malonyl-CoA is being produced, it wants to become palmitate. So أكيد it's going to prevent the breakdown of this palmitate. And why are we building and breaking at the same time? عرفتوا? First remember, CPT1 is broken down and inhibited, through malonyl-CoA, which enacts its action through insulin, so that we can have successful fatty acid synthesis. تمام? I want you to remember glucagon, AMPK, and PPAR, they increase fatty acid oxidation. I think you know this already, we explained it. CPT2, it's located in the inner mitochondrial membrane, or it does the opposite of CPT1. It removes the carnitine from the fatty acid so that it can go inside the mitochondrial matrix. Now we're going to talk about the deficiencies of CPT1, CPT2, and carnitine. We're going to start with carnitine deficiency, meaning in all of the carnitine in the body is يعني ما هو موجود. That's why we have very low total carnitine. This will lead to what? Impaired transport of long-chain fatty acids into the mitochondria. ليش? If we go back here, if we don't have carnitine, okay, CPT1 and CPT2 are working, but if there is no carnitine, يعني, again, the country will not let me in because I'm not going inside with the American citizen, as the example I'm giving you. But this is what will be impaired in carnitine deficiency. I want you to remember the clinical presentations, because the clinical presentations of carnitine deficiency is a mix of both CPT1 and CPT2. You're going to notice in the CPT1 is hypoketotic, hypoglycemic, or CPT2 is muscle pain. In carnitine deficiency, it's a mix of all of these. We have hypoketosis, hypoglycemia, cardiomyopathy, and muscle weakness. منطقي? And obviously, we said we're going to have very low total carnitine. CPT1 deficiency, this is going to be bad. Why? Because the fatty acids will not be able to enter the mitochondria. But this will lead to, of course, low total carnitine, not very low, but low. It affects the liver, or we're going to have hypoketosis, where we won't have ketone bodies. Or you might ask why. Why in these disorders we don't have ketone bodies? I expect you guys to know, but I'm going to explain it to you. Let's go here. What is the result of fatty acid oxidation, acetyl-CoA? And you guys, I think, should know. If not, we will go through it. But acetyl-CoA is the precursor of ketones. So if we're not able to break down fatty acids into acetyl-CoA, how are we going to get the acetyl-CoA to make ketones? So that's why ketones will be low. عرفتوا? But this is CPT1 deficiency. It's basically liver disease, hypoketosis, hypoglycemia. And we have to avoid fasting. We need energy because we can't break it down to make it ourselves, so we have to take it. CPT2, guys, it's very easy. Everything that has to do with muscle and all the muscles. Skeletal muscle, muscle pain, rhabdomyolysis, myoglobinuria, and exercise intolerance. If you see a scenario with all of the clinical signs are describing muscle weakness وكذا, then it's a CPT2 deficiency. خلاص? طيب. Remember how I told you guys in early on, the first lecture, إنه كان عندنا fatty acid synthase, which does these 4 reactions to make malonyl-CoA into palmitate. طيب, ترى نفس الشيء. Here, this beta oxidation is not very simple. It has its own processes. What are these processes? Here. The opposite of the ones in synthesis, which can reduction or dehydration. Here is the opposite. We have oxidation instead of reduction, hydration instead of dehydration, another oxidation, then cleavage. I'm gonna say a bunch of things. You guys have to memorize them. Oxidation number one happens through the enzyme acyl-CoA dehydrogenase. You have to understand this and know it. And oxidation number one, not number two, oxidation number one will give us FADH2. Wallahi, this came in a question once and I got it wrong. That's why I don't want you to get it wrong, so I want you to memorize it. Well, I kind of didn't see it in your past questions, so know it from me if it's not there. And oxidation number two is what's going to give us NADH. For what, oxidation number one, FADH, oxidation number two, NADH. And the only enzyme I want you to remember from here is for oxidation number one, which is acyl-CoA dehydrogenase. Why do I want you to remember this enzyme? Because it's associated with a disease. That disease is medium chain acyl-CoA dehydrogenase deficiency, which is deficiency of the enzyme we said. What will it lead to? It will lead to hypoketotic, hypoglycemic state, طيب, but with elevated carnitine. And now let me explain to you how you can differentiate between this one, the MCAT deficiency, and the CPT1 deficiency. You have to understand how to differentiate between them, ليش? Because all of them are causing hypoketosis and hypoglycemia. So we have to understand how to differentiate. Let's go here to the pathway. If CPT1 and CPT2 and carnitine are deficient, what does this mean? It means that we have low carnitine in the blood, and that's why we can't proceed with the oxidation. But when we have a deficiency of MCAD, it means we are in this step, the beta oxidation step. What does this mean? It means we successfully got the fatty acids to the mitochondria. For the carnitine is good, the levels of carnitine are normal. For the issue is in the oxidation, not in the carnitine. Well, the body will notice in all of the fatty acids are successfully making it to the mitochondria, but they're not being oxidized. ليش? For the body thinks it needs to push more carnitine. For carnitine will be a lot in the blood. It will be markedly elevated. خلاص? This is how to differentiate. CPT1, CPT2, low carnitine. MCAT deficiency, elevated carnitine. Well, the symptoms are ray-like symptoms, which basically refers to babies vomiting, being lethargic, confused, and behavioral changes. تمام? Changes in their behavior, basically like fighting, which would occur due to brain swelling. You don't need to know all the details, just a skim of this. So in this condition, the fatty acids, they enter the mitochondria successfully, but the beta oxidation is impaired because we have the enzyme deficiency. And this is the enzyme, acyl-CoA dehydrogenase. So we can't do the oxidation to continue and produce the final acetyl-CoA basically. طيب. Now I want to talk about odd-chain fatty acids. We just talked about breaking down fatty acids. Which one in specific? Palmitate, which has 16 carbons. But what if a fatty acid has 17 carbons? It's odd-chained. للأسف, we have a whole other pathway. We're going to get this fatty acid and it's going to become propionyl-CoA. It's a very simple pathway. Propionyl-CoA will become methylmalonyl-CoA. Through what enzyme? Propionyl-CoA carboxylase. And this enzyme needs biotin. So this is the second enzyme today that needs biotin. The first one, إيش كان? Acetyl-CoA carboxylase in fatty acid synthesis, acetyl-CoA to malonyl-CoA. This is the second enzyme, propionyl-CoA carboxylase, and it needs biotin to transport propionyl-CoA or transfer it بالأصح and make it methylmalonyl-CoA. Then methylmalonyl-CoA through the enzyme methylmalonyl-CoA mutase will become succinyl-CoA. And this enzyme needs vitamin B12. So if we have a biotin deficiency, propionyl-CoA carboxylase will not work, so we can't make methylmalonyl-CoA, so propionyl-CoA will accumulate. However, if we don't have B12, what's this? However, if we have methylmalonyl-CoA mutase deficiency or a B12 deficiency, we can't make succinyl-CoA, so methylmalonyl-CoA will accumulate. خلاص? وبعدين succinyl-CoA, you guys know, it's in the citric acid cycle. So that's what I want you to know about how odd-chain fatty acids are broken down and metabolized. And last but not least in this lecture, I think, we're going to talk about ketone bodies, how they are made. All I want you to know is that we start with acetyl-CoA, as I said previously. With this acetyl-CoA will become HMG-CoA by HMG-CoA synthase. This is the rate-limiting step of ketone body formation. So this is all I want you to know, is that acetyl-CoA is the starting point. the rate limiting step is HMG-CoA synthase to give us HMG-CoA. بعدين we're gonna get the ketones اللي هم acetoacetate, 3-hydroxybutyrate وهكذا. تمام؟ Now, I want you to know that tissues outside the liver, such as the brain, خصوصيا the brain, they love ketones because they know how to use them. They have an enzyme called thiophorase. The brain has thiophorase. And what does thiophorase do? It breaks down the ketones into acetyl-CoA. And the brain will use acetyl-CoA for energy. But the liver, it can't use ketones for energy. Why? Because it doesn't have thiophorase. Is this clear? The brain has thiophorase, so it can use ketones for energy, but the liver does not have thiophorase, so it's unable to use ketone bodies as an energy source. Second lecture over. We're going to move on to the questions. You can pause and answer, but this question is asking about a substrate or a molecule that regulates both fatty acid oxidation and synthesis. What is it? We already said. Malonyl-CoA. Malonyl-CoA inhibits CPT1, which will increase fatty acid synthesis and decrease fatty acids oxidation. Second question. Very long question, but what they're saying is the child has MCAT deficiency. Why would this lead to low circulating ketones? I already explained this to you guys. The patient does not release ketone bodies from the liver, because we don't have acetyl-CoA. If MCAT is deficient, it means we can't break down fatty acids, which means we can't get acetyl-CoA, which means we can't do this pathway of making ketone bodies. The ketones will be low in MCAT deficiency. تمام؟ طيب, this question is asking about how insulin inhibits fatty acid oxidation. I told you guys, I told you that there is a substance that inhibits CPT1, or there is something, or not something, that this something is pushed by insulin. It's malonyl-CoA. I mentioned it in the slides. I'm going to go back just so you guys can link it well. Here, CPT1 inhibited by malonyl-CoA through insulin. تمام? Next question is this one. They're asking about muscle disorders, muscle pain, muscle weakness, نعرف إيش skeletal muscle. It's gonna be CPT2 deficiency. I think this is the last question. You can pause to answer. What are they telling you? They're telling you that this patient has symptoms that are relieved when we give biotin. So which enzyme is deficient that when we give biotin, it becomes good? We talked about it before even giving you the answer. It's here. Propioncyl-CoA carboxylase. If the question was about B12, you'd say methylmalonyl-CoA mutase. خلاص? So the answer is propioncyl-CoA carboxylase. Next lecture, cholesterol and steroid metabolism. Very easy lecture, inshallah. حتى اللي بعدها, lipoproteins, they are very easy lectures. First, we want to understand how is cholesterol made in the body. طبعا cholesterol can be taken from outside, but believe it or not, most of the cholesterol that you, dear first year, have in your body comes from de novo synthesis. That's why it doesn't matter that much if you reduce your cholesterol intake to become healthier. Of course, it's good, but what must be regulated is the de novo synthesis, because 80% of the cholesterol in your body comes from de novo synthesis. طيب, how do we make cholesterol? The first two steps are the same one as in ketone body synthesis. Acetyl-CoA, HMG-CoA, by HMG-CoA synthase. But if we go back to ketones, here, instead of going from HMG-CoA to the ketones, now we are going from HMG-CoA to something called mevalonic acid. And how do we make mevalonic acid? Through the enzyme HMG-CoA reductase. Very important enzyme. This is the regulated, rate-limiting, committed, عمي عمكم, most important enzyme in the cholesterol synthesis pathway. تمام? HMG-CoA reductase. It will give us mevalonic acid. Then you don't have to know the rest. Just know it in the type of knowledge that when you see it, you'll know it. اللي هم the metabolites of the cholesterol pathway, which are isopentyl pyrophosphate, geranyl pyrophosphate, farnesyl pyrophosphate, squalene, and lansitol, which will eventually give us cholesterol. The most important thing I want you to know is that we start with acetyl-CoA, then HMG-CoA, and HMG-CoA through HMG-CoA reductase. The most important enzyme of this pathway will give us mevalonic acid. With this mevalonic acid will then become cholesterol. طيب, there's something I want you guys to know. إنه cholesterol in the body cannot freely move on its own. It has to be in the form of esters to move. It has to be esterified. طيب, what adds this ester على الكوليسترول? An enzyme called lecithin, cholesterol acetyl transferase. Lecithin, cholesterol acetyl transferase, sorry, LCAT. Okay, it's LCAT. For LCAT, it esterifies cholesterol so that cholesterol can move in the body better. تمام? Well, there is another enzyme that I want you guys to know, which is CETP, cholesterol ester transfer protein. What does this do? It exchanges lipids between the lipoproteins. We're going to talk about lipoproteins actually. We have HDL, LDL, VLDL. So CETP, all it does, it gives مثلا, it takes cholesterol مثلا from the LDL and it puts it in the HDL. That's all I want you to know about CETP. طيب, de novo synthesis of cholesterol, where does it happen? In the liver. And as I said, it constitutes most of the cholesterol in a human. And HMG-CoA reductase is the rate-limiting enzyme, the most important enzyme in all of this. طيب, طبعا cholesterol in itself cannot be broken down. It has to be transformed into bile salts, and then these bile salts will be excreted in urine. So just know this. It's very, very important. Now we're going to talk about the regulation of HMG-CoA reductase. Very easy, guys. Wallahi, very easy. When we are well fed, we have a lot of nutrients in the body. We have a lot of insulin. We're going to activate HMG-CoA reductase. How? By dephosphorylation, a phosphatase, removing the phosphate. So this will lead to more cholesterol synthesis. So guys, it makes sense. When your body is well fed, it has a lot of nutrients, it's like, you know what? Let me make cholesterol. كأنه واحد فلوسه زايدة. He's like, you know what? Let me travel. This is exactly what's happening here. However, when you're fasting, when you don't have energy, this isn't the time for cholesterol synthesis. When there is glucagon. لما الواحد مضفر ما عنده فلوس ما حيقول let me go travel. He's gonna focus on the necessities basically. So glucagon will stimulate protein kinase A, which will phosphorylate the HMG-CoA reductase, leading to it being inactive, leading to decreased cholesterol synthesis. Now let's move to this part, guys, of the diagram. Please memorize this like your name. It's very important. Cholesterol as a molecule can do something called transcriptional control, and it can control a lot of different enzymes. First of all, when we have a lot of cholesterol, منطقيا, logically, it's going to inhibit HMG-CoA reductase because خلاص we have a lot of cholesterol. Something else that's important, that when we have a lot of cholesterol inside a cell, we're going to stimulate an enzyme called acetyl-CoA acetyltransferase, ACAT. Does it ring a bell? Yes, it does, because it's similar to LCAT, not just in name, بس كمان in function. مو قلنا إنه LCAT will esterify cholesterol? Yeah, well, ACAT does the same, but there's just one difference. LCAT will esterify cholesterol outside the cells, in the plasma, كذا. But ACAT will esterify cholesterol inside the cells. But very important to know, when there is a lot of cholesterol, it will activate ACAT to turn this abundance of cholesterol into esters so they can be transported better. The third thing is that cholesterol will decrease the LDL receptor synthesis. What are LDL receptors? LDL receptors are receptors on cells. This is a cell. We have the LDL receptor. When this receptor is active, it takes cholesterol inside the cell. But when we already have enough cholesterol inside the cell, خلاص, why do we need this LDL receptor? That it will basically be broken down. We have decreased LDL receptor synthesis leading to decreased uptake of cholesterol. تمام? Now we're going to talk about how cholesterol could lead to heart disease. Levels of cholesterol are related to heart disease. More cholesterol we have, more chances of heart disease. But we have to regulate the cholesterol levels. How? A lot of medications, we're going to talk about them all, but the most important medication is statins. What are statins? They are analogs of HMG-CoA. They are similar to HMG-CoA, اللي هم this. What do they do? Since they are similar to HMG-CoA, هذول statins, they competitively bind to HMG-CoA reductase, leading to its inhibition, meaning we won't have a lot of mevalonic acid, so not a lot of cholesterol will be synthesized. شايفين less cholesterol synthesis. And what will this mean? إذا a lot of cholesterol will make less LDL receptors. Less cholesterol will lead to increased LDL receptors. All of this excess cholesterol in the blood will now be taken inside the cells. So we will have the clearance of lipoproteins and cholesterol. Now, another disease I want you to know other than atherosclerosis, which we will talk about in a moment, is familial hypercholesterolemia. Hypercholesterolemia, hyper a lot, cholesterol. So we have a lot of cholesterol. Why does this genetic disease happen in people? Because they have defective LDL receptors. يعني a lot of cholesterol is found in the blood, and because the LDL receptors are not working, we can't take this cholesterol inside the cells. So we have familial hypercholesterolemia. Now let's talk about bile acids. We said that this is how we excrete cholesterol. So bile is synthesized in the liver, stored in the gallbladder, and it is the major excretory form of cholesterol. You don't need to know this a lot. It's in GI mostly. But primary bile acids happen through hydroxylation, and secondary bile acids through dehydroxylation. Gallstones can result from increased biliary cholesterol excretion. So any condition of a person where there is a lot of cholesterol, this abundance of cholesterol will mean we will have a lot of bile acids. Too many bile acids can give us a condition which is gallstones. So how do we treat gallstones? Cholecystectomy. Memorize this like your name. It came in a past question before. How do we treat gallstones? Cholecystectomy. طيب. Now we're going to talk about خلصنا talking about general information about cholesterol and how to make cholesterol. Now I'm going to talk to you guys about something called steroidogenesis. What is steroidogenesis? It means creating steroids. Like, why is this pathway in this lecture? Because steroids are created from cholesterol. But that's why it's in this lecture. Like, first of all, the first step of the reaction, which is actually the rate limiting step, is bringing cholesterol from the cytoplasm into the mitochondria. Because cholesterol, for it to become a steroid, it has to be in the mitochondria. So this is the rate limiting enzyme, the rate limiting step. It is the first step, which is the rate limiting one. What is the enzyme called? STAR. وانتوا كلكم نجوم. Anyone listening to this right now. The enzyme is called steroidogenic acute regulatory protein. So this STAR enzyme will bring cholesterol from the cytoplasm to the mitochondria. That is the rate limiting step of steroidogenesis. Remember this, okay? Then the next step of steroidogenesis is changing cholesterol into progesterone. Well, then progesterone will have three different pathways because three different steroids are going to be produced from progesterone. Either androgens, testosterone, estrogen, for sexual features, or glucocorticoids, which is cortisol, or mineral corticoids. This is the pathway I want you to focus on. Mineral corticoids are aldosterone, which why is aldosterone important? For sodium and water retention. So let's say a person doesn't have enough aldosterone. What will happen? Salt wasting. The salt will be excreted instead of retained. Like, two important enzymes I want you to know. 21 alpha hydroxylase, which changes progesterone into 11-deoxycorticosterone. I also want you to remember this, deoxycorticosterone. And another enzyme, which is right after it, 11 beta hydroxylase, which takes the deoxycorticosterone and changes it into corticosterone. Something important I want you to know, 11-deoxycorticosterone has the functions of aldosterone, but corticosterone doesn't, okay? Why is this important? Because we're going to talk about the deficiencies of both of these enzymes. Very important for you to know. Let's say 21 alpha hydroxylase is deficient. What will happen? Progesterone can't become deoxycorticosterone. So what will this lead to? It will lead to not having aldosterone because this whole pathway is blocked. So decrease aldosterone and decrease deoxycorticosterone. وكمان we will have decreased cortisol because usually this progesterone is 33% here, 33% here, 33% here. When this is blocked, what will happen? This will actually become 25% and most of the progesterone will become testosterone and estrogen. عرفتوا? That's how this deficiency will impact cortisol. Everything in 21 alpha hydroxylase deficiency will go down, except for what? Androgens. They will go up. زي ما انتم شايفين هنا, androgens will go up. So what will this lead to? Decrease aldosterone, salt wasting, important. I told you about it. Decrease cortisol, fatigue and hypoglycemia, and what will increased androgens lead to? Virilization, which is basically formation of male characteristics in females, and precocious puberty, hirsutism, and acne. And 21 hydroxylase deficiency is often referred to as congenital adrenal hyperplasia. تمام? So this is 21 hydroxylase deficiency. Next, we have 11 beta hydroxylase deficiency. It's different. Why? Because instead of inhibiting the pathway here, the pathway will be inhibited here. So the deoxycorticosterone can't change into corticosterone. This is better than 20 alpha hydroxylase, by the way, because at least we have deoxycorticosterone. And what did I tell you guys about deoxycorticosterone? Is that it has the effects of aldosterone. So yes, aldosterone will be low, but because the oxycorticosterone will be high, we're going to have hypertension and hypokalemia. Yani we're going to have the effects of aldosterone, but not through aldosterone, through deoxycorticosterone. And we're going to have too much of it to the point where we're going to have hypertension because of a lot of fluid retention. تمام? But like in 21 alpha hydroxylase deficiency, the rest is the same. Cortisol will be decreased, while androgens will be increased. تمام? For the same consequences as 21 alpha hydroxylase deficiency, but there's one difference that we have increased deoxycorticosterone, which would lead to hypertension. تمام? I hope this is clear. Very important slide. Memorize it like your name. والله مجد. طيب. Now we're going to move on to steroids. We're gonna say in the steroid hormones, they are not water soluble. خلاص, just memorize it. It has to be bound to globulins to be transported. فcorticosteroid مثلا binds to globulin, which carries cortisol. Sex steroid binding globulin will carry testosterone. Not very important, just know it for your knowledge sake, يعني, basically. As we said, the rate limiting step in steroid synthesis is what? Taking the cholesterol from the cytoplasm to the mitochondria, through which enzyme? SAR, steroidogenic acute regulatory protein. طيب. Something I want you guys to know, in clinical settings, in the labs or clinics, how can we know someone has 21 hydroxylase deficiency other than the signs we talked about? They would have increased plasma 17 hydroxyprogesterone. Just know this as a fact because it could come as a question. طيب, checkpoint. This lecture is over. We're going to move on to the questions. So we want to explain why statin therapy is effective for people with a lot of cholesterol. The reason is that they inhibit HMG-CoA reductase. This question is talking about which statement best describes the condition of congenital high cholesterol. We said it. Why does familial hypercholesterolemia happen in the first place? I'm going to go back actually because I want you guys to see it for yourselves. Due to defective LDL receptors. But we can't take in the cholesterol in the cells, so the cholesterol will build up where? The blood. Exhibits a deficiency of LDL receptors. Next question. A baby has 21 hydroxylase deficiency. What would you expect? Hypopigmentation? Did I ever mention this? لا, you don't choose it. Salt stature? Did I ever mention this? ما عليكم منه. Hypernatremia? Did I ever mention this? I did, but hyponatremia. Here's the trick. Because in adrenal 21 hydroxylase deficiency, we will have less aldosterone, which means what? Aldosterone, what does it do? It preserves sodium. So in hypernatremia, we have the sodium. But لا, here we don't have aldosterone, so we will have hyponatremia. But the answer will be salt wasting crisis, because we are wasting the salt. Hyponatremia. Next question. The regulatory step of steroid synthesis happens through which molecule? We said steroidogenic regulatory protein. Next question. It's basically asking you, how does glucagon يعني, affect the synthesis of cholesterol biosynthesis? The answer will be that glucagon will phosphorylate HMG reductase, CoA reductase. When we phosphorylate it, you inactivate it. طيب. Now we're going to go to the next lecture, which is lipoproteins. Very easy lecture, 2 and 1. So wait, I simplified them for you. You're going to enjoy this lecture, inshallah. طيب. First of all, what are lipoproteins? Lipoproteins are lipids and proteins, lipoproteins من اسمها. What is their function? Transporting and carrying different substances in the body. As simple as that. We have 5 different types of lipoproteins that I want you guys to know. Chylomicrons, VLDL, which is very low density lipoproteins, IDL, intermediate density lipoproteins, LDL, low density lipoproteins, and last but not least, HDL, high density lipoproteins. And these are the 5 different types of lipoproteins, and each one carries something different. We're going to go through this again, but if you memorize this from this slide, ضيعتوا على نفسكم. Chylomicrons, triglycerides, full stop. VLDL, triglycerides, full stop. We're going to tell you the difference بعدين between the triglycerides of chylomicrons and VLDL. There is a huge difference and you have to memorize it, but let's keep it for later. IDL, I don't care about it. LDL, cholesterol. Well, cholesterol is bad, so LDL is bad. As simple as that. HDL is good. Why? Because it doesn't have a lot of cholesterol. This is what I want you to remember about HDL. خلاص? I hope what I said is clear. Now I want to explain a concept that you will definitely get at least one question about, which is the size of the lipoproteins relative to their density. What if I tell you guys that you won't even need to memorize this? I'm gonna make it logical for you. Let's speak physics. When something is very dense, what does it mean? It means that it is very concentrated relative to its size, meaning that the smaller something is, the higher the density will be if the concentration is same, صح? فإن a high density lipoprotein, it's the highest density, so أكيد it will be the smallest because it has the most concentration of protein. بعدين we have very low density lipoprotein, it will be the second biggest, it will be big, لأنه it's very low density. Chylomicrons ما في density باسمه أصلا. This thing doesn't even have a density, so it's so freaking fat. IDL or LDL, LDL is second smallest to HDL, then we have IDL. So this is an easy way to remember the sizes. Chylomicrons biggest, VLDL, then IDL, then LDL, then HDL. With the density is the other way around. طيب. I want you guys to know إنه lipoproteins مو هم lipolipids and proteins. طب we talked about the lipids. What about the proteins? These are the proteins. There are 5 different types of apolipoproteins, the proteins that are found on the lipoprotein surface. 5 types, you need to know them, you need to memorize them. We will talk about them in the slides بعد شوي, but again, I'm gonna explain them all in this slide to give you a very good basis. We have ApoB, ApoC2. Does ApoC2 ring a bell? It does ring a bell because it activates lipoprotein lipase. You guys should know this. Well, matter of fact, I'm gonna go back because I want you guys to see it for yourself. Do you guys remember? Fatty acid oxidation, lipoprotein lipase with breaks down tag is activated by what? By apoprotein C2. Just wanted to leave you with that. Let's go back. طيب. For ApoC2, we already know what it does. It activates lipoprotein lipase to break down the triglycerides, the chylomicrons, and VLDL. Why those two specifically? Because I told you guys, the triglycerides are found in chylomicrons or VLDL. Then we have ApoE, ApoA, and ApoC3. C3 is similar to C2, صح? In name and in function. The difference is that C3 will inhibit lipoprotein lipase. For that's 2 of the lipoproteins, خلاص, we already explained them. Now we have ApoB. ApoB has 2 different subtypes, ApoB48 and ApoB100. Why does this division happen? Why do we have 2 different subtypes? Because of RNA editing. Remember this. You will be asked about this probably. So ApoB48 is found on chylomicrons. That's what I want you to know. ApoB100 is found on VLDL and IDL as well. But so this is the first difference between chylomicron and VLDL. Chylomicron has ApoB48 while VLDL has ApoB100. Well, ApoB48 of the chylomicron is responsible for intestinal matters, while the ApoB100 of the VLDL is responsible for liver, hepatic matters, okay? Now let's go to ApoE. ApoE is very simple. If anything wants to be taken up by a liver, it has to have ApoE. بس, as simple as that, تمام? Then we have ApoA. ApoA is the big boss. It's my favorite one. Because it is the major protein of HDL. What is HDL? We said it's the good lipoprotein that has very little cholesterol and a lot of protein. Now, what is the function of ApoA? Simple. It activates ALCAT and it's involved in a reverse cholesterol transport. We're going to talk about this. In reverse cholesterol transport, it's an important function of HDL and ApoA. ApoA is in the HDL. What is reverse cholesterol transport? As you guys know, most of these lipoproteins, you're going to find out in their function is taking cholesterol from the liver to the tissues of the body. This is bad. They are just depositing the cholesterol everywhere. But HDL is the only one that does reverse cholesterol transport. It takes the cholesterol from the tissues back to the liver. It cleans your blood, basically. I hope this slide is clear. Now, basic information, triglycerides, phospholipids, cholesterol, and cholesterol esters, all of these substances and proteins, they are transported in lipoproteins. They are the carriers. What are the lipoproteins? Chylomicrons, VLDL, IDL, LDL, and HDL. طيب. Now, the core of the lipoprotein, what is it made of? You don't need to know this, just basic information. It's made of tag and cholesterol esters, or the surface is made of apoproteins. We said this already as the surface here, apoproteins. With the polar groups face outwards, making lipoproteins soluble in plasma, not very important. What I want you to know is if someone asks you to calculate the total cholesterol in a human, you will add LDL plus HDL plus VLDL. خلاص? The cholesterol in the LDL, the cholesterol in the HDL, and the cholesterol in the VLDL. طيب. Do you guys remember what I said? إنه VLDL has triglycerides و LDL has cholesterol. I already said this. So this means if we have a condition of hypertriglyceridemia, it means we have a lot of VLDL. And if we have hypercholesterolemia, it means we have a lot of LDL. Makes sense, ولا? طيب. Do you guys remember how we said in a VLDL is larger than LDL? If we go back here. Yes, it is. But just a small fact, not very important in my opinion, but حطيته for completion's sake. VLDL, it travels faster in the electrophoresis. It's faster than LDL, even though it's bigger. Why? Because it has more apoproteins. Simple as that. طيب, let's talk generally about apoproteins. They are lipid-binding proteins that are on the lipoproteins. What do? They have functions and they decide the fate of this lipoprotein and what it will end up doing. تمام? طيب, we said ApoB has two subtypes. ApoB48, which is produced in the intestine for the chylomicrons, and ApoB100, produced in the liver for meal VLDL. The different subtypes result from RNA editing. As we said, lipoprotein lipase, we already know this, guys. It's activated by ApoC2 or deactivated by ApoC3. You see, guys, this rectangle, it's just one line. So, wallahi, a question came about it once. So I want you to memorize this line. I won't even say it. Just memorize the line عشان ما نزعل من بعض. I'm gonna go to explain this. IDL can be converted into LDL. How? By an enzyme called hepatic lipase. وصلى الله عليه. طيب. Let's talk about the first lipoprotein, which is chylomicrons. What is the function of chylomicrons? They transport dietary triglycerides, which come from the outside, from the small intestine to peripheral tissue. Guys, wallah, it's not autism. The reason why I'm emphasizing dietary small intestine is because in VLDL, we also transport triglycerides, but VLDL will transport endogenous triglycerides, the ones made in the body, and not from the small intestine, from the liver. خلاص? Do you guys see the difference? Wallah, very important to differentiate. For chylomicrons, dietary triglycerides from small intestine to peripheral tissue, and it appears in a plasma after a fatty meal. You have to eat something from outside that's fatty, or that's how chylomicrons will appear. طيب, chylomicrons, when they are in the plasma, they will cause the serum to look milky, turbid, white, okay? As we said, chylomicrons have ApoB48, which is required for the secretion from the small intestine to take it to the peripheral tissue, and it acquires, not contains, it takes, acquires ApoC2 and ApoE from where? From HDL. ApoC2 will cleave the chylomicron when needed, or ApoE will allow the chylomicron to go to the liver if needed. Or something I want you to know from now, HDL is the one that donates the ApoC to ApoA. Kareem, what did we say HDL has that's very important? ApoA. which is activates LCAT to acidify cholesterol and involved in reverse cholesterol transport, cleaning. طيب, I'm gonna skip HDL, VLDL, since it is the counterpart of chylomicron. VLDL transports endogenous triglycerides from the liver to the peripheral tissue, and it contains what? ApoB 100, not 48. بس كمان it acquires ApoC2 and ApoE from HDL. طيب, VLDL is hydrolyzed by lipoprotein lipase, which will become IDL. It's the first remnant. IDL, some of it is taken by the liver, while the rest turns into LDL. How? By the enzyme hepatic lipase. We already talked about this. Let's go back to HDL. Very important. What is the function of HDL? It transports cholesterol from peripheral tissue to the liver, reverse cholesterol transport. That's a very good thing. That's why HDL levels are inversely related to the risk of atherosclerosis, meaning the more HDL we have, the less risk of cardiovascular diseases. طيب, if someone wants to have a lot of HDL in their body, what should they eat? Fish and vegetable diets. طيب, how does the liver take up cholesterol from HDL? HDL, مو احنا قلنا HDL will take the cholesterol from the tissues and will present it to the liver. طيب, the liver can't take this cholesterol إلا through a certain receptor. It's called scavenger receptor class B type 1, SRB1. Memorize it, very important. This is the receptor that the liver will use to take the cholesterol from the HDL. تمام, SRB1. طيب, as we said, HDA has ApoA, which activates LCAT, and it donates ApoC2 and ApoE to others. CETP. Okay, now I'm gonna talk to you about a bad guy. The bad guy is CETP. Why is it a bad guy? Do you guys know how HDL takes the cholesterol and puts it in the liver? HDL, while it's going to the liver, it will be met by CETP. CETP will be like, you know what? Give me your cholesterol, take triglycerides instead. What will this lead to? It will lead to the HDL having triglycerides instead. فحيروح الليفر with no cholesterol. We didn't clean up anything. ويا ليت the CETP will clean anything. It won't. It will give the cholesterol to VLDL. VLDL doesn't help in cleanup. Well, guys, if you remember, we already talked about CETP before. I told you guys that its function is to exchange molecules between lipoproteins. This is the exchange we're talking about. For CETP is not good. The increased CETP is associated with lower HDL levels, which means higher risk of atherosclerosis. طيب, we did this. Now we have LDL. LDL is literally the opposite of HDL. إذا HDL بيساعد ينظف, LDL بيوسخ الدنيا. It's causing a mess. It delivers cholesterol to peripheral tissues, literally giving the peripheral tissues cholesterol. And it expresses ApoB 100. Now, LDL, how is it metabolized? You don't need to know this, just for completion's sake. It binds to clathrin-coated pits, which will be endocytosed, then broken down in lysosomes. Now, I want to talk about ACAT. This isn't related to LDL exactly, but I put it in this section. Do you know how LCAT قلنا it esterifies cholesterol outside the cell? It's the function of ApoA in the HDL. ACAT also converts free cholesterol to cholesterol esters, but intracellularly. We already said this, بس حطيتها for completion. Something very important I want you guys to know. A transcription factor اسمه steroid regulatory element binding protein 2. SREBP2. I'm going to summarize it as in a very simple way. What does this do? It's inside a cell. It detects the cholesterol levels. When the cholesterol levels get too low, this transcription factor, SREBP2, will be activated. What will this lead to, this activation? It will lead to increased LDL receptors to take more cholesterol. And it's only activated because the cholesterol in the cell is too little. So we're going to activate the LDL receptor to take in cholesterol. That's one. And two, activate HMG-CoA reductase gene to make more cholesterol. Right, now I'm going to talk about atherosclerosis, the disease that is caused by cholesterol. تمام? I'm going to explain to you how it happens. It all starts with having too much cholesterol inside the cells. When we have too much cholesterol inside the cells, what will happen? I already told you guys من زمان. إنه the cholesterol itself. Do you remember, guys, the diagram that I begged you to memorize? إنه cholesterol can regulate its synthesis by either inhibiting LDL receptors ولا ACAT ولا The third one, what was it? Let's go back to it actually, because it was very important. You have to memorize it. It was mm, here. Inhibits HMG-CoA reductase أو stimulates ACAT أو decreases LDL receptor synthesis. This is how cholesterol does feedback. So in atherosclerosis, we have too much cholesterol in the cell, which will lead to cholesterol inhibiting the LDL receptors. So we will no longer have receptors that take the outside cholesterol. What will this lead to? It will lead to the LDL cholesterol that is outside the cell to build up and increase, which is all fun and games, but there is a problem. When cholesterol is inside the cell, it is safe, it is esterified. But when it's outside the cell, it's not safe. It gets oxidized. It enters arteries and gets oxidized by reactive oxygen species. So it becomes oxidized LDL. And there is a problem. Macrophage واحد ملهوف. It has a certain receptor that's macrophages. They have certain receptors called scavenger receptor class A. SRA, subhanallah, their only task in the world is to take up specifically oxidized LDL. But when too much oxidized LDL builds up, this macrophage will come and through the scavenger receptor class A, it will take up this oxidized LDL. What will this lead to? Cholesterol accumulating in macrophages. طيب, is that bad? Of course it's bad because we will have foam cell formation. Foam cells are what will end up blocking the arteries, leading to atherosclerosis. Guys, sclerosis means thickening, hardening. Atherosclerosis is basically thickening in the artery. So we have here thickening or narrowing of the artery because of the foam cell deposition. خلاص, I want you to memorize and understand this whole pathway for atherosclerosis. طبعا, something important I want you to know. You might ask, طيب, does this happen in everyone? Not specifically. It happens in people with an abnormal LDL. What is the apoprotein that's usually in LDL? We already said it here. Gonna go back. Apo B100. In people that get atherosclerosis more likely or more often, they don't have the normal Apo B100. They have a defective one. اسمه lipoprotein A. Lipoprotein A is basically ApoB100 and a modification. The final product is called lipoprotein A and it leads to atherosclerosis. Let's talk about atherosclerosis. When intracellular cholesterol increases, LDL receptor decreases, which leads to LDL being oxidized outside the cell by reactive oxygen species. We already said this. Then the macrophage, which have SRA, the receptor, scavenger receptor class A, will take up this oxidized LDL. Cholesterol accumulates in the macrophage. It makes foam cells. Foam cells will accumulate in the arteries, leading to narrowing of the arteries and atherosclerosis. So as we said, lipoprotein A, it is the mutant form of the ApoB100 that is found in LDL that is bad, which causes atherosclerosis. So high LPA, lipoprotein A, increases cardiovascular disease risk. طبعا lipoprotein A is different than apoprotein A. Apoprotein A حق ال HDL. This is lipoprotein A, something different. طيب, now we're going to talk about medications that are used to treat hyperlipidemia in general. Okay? We already talked about statins, but I'm not going to talk about them more. خلاص, we know what they do exactly. We're going to start with cholestyramine. What does cholestyramine do? Do you guys know how bile acids are basically how cholesterol is excreted? Now, the thing about bile acids is that after they do their jobs, some of them are excreted, but the others are reabsorbed. When they're reabsorbed, it means the body doesn't need so many bile acids. ليش؟ لأنه خلاص we are recycling the ones we have. فcholestyramine, they will prevent the reabsorption of bile acids. But all of the bile acids get excreted. What will this lead to? It will lead to the body saying, oh, we don't have bile acids. Oh, we need to make more bile acids. So what will happen? The liver will convert more cholesterol into bile acids. And this will lead to decreased cholesterol in the liver. Okay? Next, PCSK9 inhibitors. Very important. They are medications, inhibitors that inhibit PCSK9. What does PCSK9 usually do? PCSK9, guys, it usually, يعني, I don't know how to explain this to you, but usually it inhibits the formation of LDL receptors. It degrades it. When we have PCSK9, we are inhibiting the LDL receptors from being formed, which means in all of the cholesterol is stuck in the plasma and can't be taken in. However, when we inhibit PCSK9, it gives us a chance to have more LDL receptors. Now nothing is inhibiting it, so it will take up the LDL or cholesterol that is making a mess in the blood or stored in the cells and acidified. تمام? What key thing about PCSK9 inhibitors that no other medication has, is that it lowers lipoprotein A. So it lowers LDL and lipoprotein A. So it even helps with atherosclerosis. Next, we have vitamin B3. What does it do? Simple. Inhibit CETP. CETP, what did it used to do? I told you guys, it's bad because it takes the cholesterol from HDL, gives it to VLDL. Now it's inhibited. فخلاص, the HDL can freely go to the liver and clean up the cholesterol. Last but not least, ezetimibe. Very simple. It inhibits the absorption of cholesterol in the intestine. That's all you need to know. Now, next, we're going to talk about the disorders and diseases that are linked with having too much lipoproteins or too little. These are the three diseases we're going to talk about. Type 1 hyperlipoproteinemia and type 2 hyperlipoproteinemia. Type 1 is about chylomicrons. We have hyperchylomicronemia. We have too many chylomicrons. Type 2 is about hypercholesterolemia. We already talked about it, the familial hypercholesterolemia due to a defect in LDL receptors. We already talked about this. So I'm going to focus on the hyperchylomicronemia, which is خلينا نقول it's a result of impaired breakdown of chylomicrons. احنا مو قلنا guys انه chylomicrons will take ApoC2 from the HDL to be broken down by lipoprotein lipase, which is only activated by ApoC2. طيب, when this is deficient, when we don't have LPL or ApoC2, the chylomicrons, how are they going to break down? They can't. They can't break down. They can't cry and have a mental breakdown, so they're going to build up. When they build up, they're going to cause hypertriglyceridemia, eruptive xanthomas, which are basically fatty lesions, and recurrent pancreatitis. That's all I want you to know. Last is A beta lipoproteinemia. What is A beta lipoproteinemia? A, when it starts in a disease, it means we have an absence of something. Beta lipoproteinemia is the apoproteins. For A beta lipoproteinemia is absence of the beta apoproteins اللي هم B100 and B48. What will this affect? Apo B100 will affect obviously the VLDL, or Apo B48 will affect the chylomicrons. فالchylomicrons won't be able to be released from the enterocytes, or VLDL will not be able to be released from where hepatocytes. What will this lead to? Very low serum triglyceride and cholesterol. Because, guys, do you remember the functions of VLDL or chylomicrons? It's to take the triglycerides after being released in the intestines and distribute them to the tissues. But if the chylomicrons can't be even released from the place where they are, اللي هم liver والintestine, how will they do their actions? عرفتوا? But obviously, as we said, the defect is absence of ApoB100 and ApoB48. I hope this summary is good. Last but not least, I will not explain this because it's everything I said, but I did it as a summary. Yani after you guys watch my past session or review my slides, you can only review this slide. It has everything that I talked about in 4 tables, which has everything you need to know, inshallah, genuinely. I won't explain it just because it's literally everything I already explained. This is only for the sake of your review, for you to have a summary. And that brings us to an end to this lecture. Practice questions. What lipoprotein causes milky appearance? Chylomicrons. HDL donates and delivers 2 apolipoproteins. What are they? C2 and E. طيب, what is the therapeutic rationale for using cholestyramine? We talked about it. Cholestyramine, it binds to bile acids, prevents its reabsorption. And if it prevents its reabsorption, what will this mean? No more cholesterol will be lost and degraded into bile acids. For this means we will have a lot of more LDL receptors to take up cholesterol because cholesterol is becoming low. For the removal of the bile acids will remove the negative regulator of LDL receptor synthesis. And I know the wording of this question is a bit weird, but I'm sure you guys understand the concept. What is the principal receptor that takes up the modified LDL in macrophages, scavenger receptor A. Lipoprotein A is covalently bound to which one? يعني lipoprotein A basically ruins which lipoprotein? We said ApoB 100. When HDL and VLDL are incubated together, what enzyme is responsible for the transfer of apoproteins? I told you guys. What enzyme takes stuff from HDL and gives it to VLDL? CETP. طيب, we're talking about a boy that was placed on a diet extremely low in fat, when basically the boy has severe, let's say, milky appearance in the serum, and triacylglycerol levels increased. The deficiency of the condition that we're talking about here is ApoC2. This is type 1 hyperlipidemia, hyperchylomicronemia, which is caused by a deficiency of apoprotein C2, leading to inability to break down chylomicrons, which leads to a lot of triglycerides and a lot of milky appearances. تمام? طيب, this question, it's talking about blockage of a major artery. We're talking about يعني atherosclerosis. Well, the question is asking what is the initiating factor of atherosclerosis? We said oxidized LDL. All right, let's continue now. We're gonna move on to the lecture, which is disposal of nitrogen. And why do we care about nitrogen? Why do we care about how it's getting degraded? Because nitrogen is a key element in amino acids. So we basically want to know how amino acids are broken down. What will happen to this nitrogen? How will we get rid of it? It's actually a very simple process. تمام? Transamination is the first step, بعدين oxidative deamination. What is transamination? Transamination is basically, مو صح, we have an amino acid. You guys know an amino acid has the carboxyl amino group, ما أعرف إيش. Transamination is simply removing the amino group and giving it to an alpha ketoglutarate. بس يعني إحنا we want to break down the amino acid, صح? We need to break it down حبة حبة. We remove the amino acid groups. But the first step is removing the amino group. The amino group will be given to alpha ketoglutarate. تمام? What will they do together? The amino acid, the amino group with alpha ketoglutarate. They will combine and give us glutamate. Keep this for later. Now my focus is about transamination. This is the amino acids. We are removing the amino acids. طيب, transamination happens through two very important enzymes. Remember them now because we will talk about them again and again. Alanine transaminase and aspartate transaminase. Those are the two enzymes that do the transamination. We will talk about them actually. طيب, فبعد transamination we said the amino group will go to alpha ketoglutarate and they will make glutamate. طيب, بعدين we said we want to get rid of everything. The glutamate will turn into ammonia. It will turn into what? Ammonia by the enzyme glutamate dehydrogenase. I want you to remember this enzyme. خلاص? What is the aftermath of what I talked about? The aftermath is simple. We have an amino acid. The amino group went with alpha ketoglutarate and gave us ammonia. طيب, what about the remaining amino acid that had an amino group taken away from it? It will be called carbon skeleton, an alpha keto acid, and carbon skeleton. Or this carbon skeleton will go into energy pathways. It can be used يعني, to make energy, glucose, ketones, وهكذا. The ammonia, طيب, where will it go? To the urea cycle to get excreted. خلاص? By transamination, remove the amino group from the amino acid. The amino group goes with alpha ketoglutarate to make glutamate, and glutamate through glutamate dehydrogenase becomes ammonia. That's about the amino. The rest of the amino acid without the amino group is the carbon skeleton, which will go into energy pathways. So amino acid catabolism, unlike fats and carbohydrates, amino acids can't be stored in the body. They have to be broken down. So catabolism involves removal of the alpha amino group, which is transamination, وبعدين oxidative deamination, which forms two things, the ammonia and the alpha keto acid, which is the carbon skeleton. So ammonia, it is the most important route for disposing nitrogen. Why? Because the ammonia will go into the urea cycle وبعدين excreted into the urine. عرفتوا? Ammonia is excreted, as we said, through the urea cycle or urine, while the carbon skeleton enters energy pathways. Oxidative deamination reaction occurs in direction to form ammonia after meals. So when we eat a lot of meals, we have a lot of amino acids, a lot of glutamate is being formed. we will do more oxidative deamination. So more ammonia will be formed basically when we're eating. Tamam, that's all you need. But carbon skeletons, they are broken down when we need energy. مو احنا قلنا carbon skeleton can be used to make energy فخلاص they're broken down into energy when the body is low in energy. طيب, the transaminases, the enzymes that do transamination. I told you guys I wanted you to remember this like your name. First of all, the enzymes of transamination اللي هم alanine aminotransferase, aspartate aminotransferase all lead pyridoxal phosphate. It comes from vitamin B6. So if you see an MCQ, you want to look for pyridoxal phosphate. If it's not there, you choose vitamin B6. Both of them are there, you choose pyridoxal phosphate. تمام? So the first one is aspartate aminotransferase. Guys, you see this line? Memorize it like your name. Wallah, you're going to get questions about it, okay? Aspartate aminotransferase equals aspartate oxaloacetate. خلاص, just remember it. Alanine aminotransferase equals alanine and pyruvate. All of them will have alpha-ketoglutarate. But you have to know that aspartate aminotransferase will give us oxaloacetate from aspartate, while alanine aminotransferase will give us pyruvate from alanine. Very important. Wallahi, I've seen questions about this. طيب. So the amino group combined with the alpha-ketoglutarate, as we said in the first slide, will give us the glutamate. Glutamate is a temporary storage form of nitrogen. وبعدين this temporary storage form of nitrogen through glutamate dehydrogenase will be broken into the free ammonia. طبعا something I don't think it's important, but it's good to know. Glutamate dehydrogenase needs NADP to work. خلاص? طيب, now we're going to talk about an important concept. You're going to get probably 2 questions about it, but it's very easy. يعني والله free grades, guys. Nitrogen balance. Nitrogen balance is basically the nitrogen in the body. If it's positive nitrogen balance, it means we are storing more nitrogen than we are losing. Negative nitrogen balance means that we are losing much more nitrogen than we are gaining. طيب, I'm gonna give you an easy way to remember negative nitrogen balance. Let's say you guys have a jar of خلينا نقول pickles. If this jar didn't have the cap, الغطاء, وطيحتوه, will the pickles fall off or will they be stored? They will fall off. We are losing them. طيب, treat your body in the same way. When our body is opened up, ما في غطاء, everything will fall off. The nitrogen will be excreted more than it's being stored. طيب, what do I mean by البody ما عنده غطاء? The body is damaged. Burns, they expose the skin. Surgery exposes the skin. Trauma could expose the skin. Illnesses doesn't directly expose the skin, but it exposes you and your immune system. Malnutrition, same things. For all of these, they will lead to losing nitrogen more than we're storing them. However, positive nitrogen balance is basically when we have a lot of anabolism. What does this mean? A lot of growth. Childhood. You're still a child, not you, but a child in general. They're growing. They need a lot of nitrogen. Pregnancy. Subhanallah, a woman has a whole other living being inside of her. طبيعي, she's going to need a lot of nitrogen. Recovery from an illness. That's different from illness. In an illness, you are losing nitrogen because you are still suffering. But recovery from an illness means إنه خلاص, you're starting to get back to normal, and this is where you need a lot of nitrogen. So you can go back to normal. And basically any time with a lot of what? Tissue growth. Now we're going to talk about a condition that's called cystinuria. Okay? Something I want you to know is that free amino acids, إحنا when we digest food, there will be free amino acids. These amino acids are taken up into the intestine by sodium-dependent secondary active transport. Remember that, حلو? Well, there are many other systems. One of these systems is responsible for taking up cysteine and ornithine and arginine and lysine. These 4. Call as a mnemonic. Hello? In cystinuria, this specific system is defective. For these 4 amino acids, cysteine, ornithine, arginine, and lysine will not be able to be absorbed. We have 2 types. Type 1, mild cystinuria. It's mild. فصح, that system is defect, but we won't find these 4 amino acids alcohol in the urine. But type 2 and type 3 are severe cystinuria. The coal amino acids will be found in the urine. تمام؟ طبعا إيش المشكلة؟ إنه when we have a lot of cysteine that's not being absorbed, it's building up, we're gonna get إيش? Kidney stones. طيب, heart nub disease, another disease. This disease comes from the defective absorption of tryptophan. Tryptophan is an amino acid, you guys know it. The issue is that when we don't absorb tryptophan, we lose it in the urine, we lose its important function, which is making vitamin B3. Tryptophan is what makes vitamin B3, so we lose this function. Patients will have pellagra-like symptoms. What are pellagra-like symptoms? Basically, niacin deficiency, B3 deficiency, which is dermatitis, dementia, and diarrhea. طيب. Okay. Ubiquitin. What is ubiquitin? Ubiquitin is basically something that goes to an amino acid and tags it. Why tags it? For destruction. It doesn't destroy the amino acid, it just tags it. It goes around and be like, okay, you're tagged, you're tagged, you're tagged, you're going to be destroyed. Hello? This is not important, but just to know how ubiquitin exactly works. And ubiquitin has glycine. The C terminal of this glycine will covalently bind to whatever amino acid has to be degraded. But again, it doesn't degrade anything. It just tags it. What will degrade the tagged amino acids? The proteasome. But the proteasome will spare ubiquitin. It will only degrade the tagged proteins. Energy for degradation is provided by ATP hydrolysis. Good to know. Fate of ammonia, very important. مو احنا قلنا glutamate dehydrogenase to ammonia. Ammonia, its fate is going into the urea cycle. But how can ammonia go to the place where the urea cycle happens? It has to be transported by something. It is transported as glutamine. Why? Because it is a non-toxic carrier of ammonia. Ammonia is toxic, but when it's transported as glutamine, it becomes non-toxic. Remember this. It comes as an example. What is the non-toxic carrier of ammonia? Glutamine. Glutamine synthetase basically will add the ammonia to the glutamate and make glutamine. فأنتوا فهمتوا من وين جاء الجلوتامين؟ مو احنا we have the glutamate broken down to give ammonia. This ammonia will combine with the glutamate and that's how we get glutamine. By what? Remember, the enzyme glutamine synthetase. طيب, I want you to know something called the glucose-alanine cycle. All I want you to know is that it is for skeletal muscles to eliminate nitrogen through alanine by the enzyme alanine aminotransferase. That's all I want you to know. Skeletal muscle, eliminate nitrogen. Alanine aminotransferase. طبعا ammonia has to be excreted through the urea cycle because if it's elevated, it is toxic to the brain. Hyperammonemia causes death and cerebral edema and convulsions. It's terrible. All right? Checkpoint. This lecture is done. Easy, inshallah. So patients with heart valve disease, we know it. المشكلة بالتفان. What can be produced by oxidative deamination? We told you glutamate. And حتى if we want to go back here, oxidative deamination glutamate, which then becomes ammonia. Well, obviously, ammonia combines with glutamate to become glutamine. But this question, nitrogenous waste from skeletal amino acid catabolism, they literally said skeletal amino acid catabolism. Nitrogenous waste. Getting rid of nitrogen, skeletal muscle. What comes to mind? Alanine, the glucose-alanine cycle. طيب, what individual would have a positive nitrogen balance? Why? كيف no protein diet? This is malnourishment, basically. Malnourishment, severe burns, chronic starvation. It's healthy child. They need to grow, so they need this nitrogen. Which of the following amino acids binds ammonia for transporting and storing it in a non-toxic form? Glutamine. We know glutamine is what stores it in a non-toxic form. طبعا, if I'm being honest, the question is a bit inaccurate. لأنه what binds ammonia, it's going to be glutamate, and then it becomes glutamine. But in this question, the answer is glutamine فعلا because it stores it in a non-toxic form. Now let's move on to the urea cycle, which will be a very easy lecture, inshallah. I summarized it in just one slide. Everything in one slide. But then obviously we're going to talk about the conditions of كذا, but everything you need to know is genuinely in this one slide. First of all, I want you to understand in the urea cycle happens in two places in a cell, the mitochondria and the cytoplasm. It starts in the mitochondria. What happens? شفتوا الأمونيا, مو the ammonia is what goes into the urea cycle. Okay, so we're going to start with ammonia أكيد, which is here. Ammonia will combine with bicarbonate and ATP to form something called carbamyl phosphate. This is the first step of the urea cycle. What is the enzyme that does this? Carbamyl phosphate synthetase 1. من اسمها باينة. Something very important I want you to know about this enzyme, that it is regulated by one thing, N-acetyl glutamate. Don't ask questions. N-acetyl glutamate regulates carbamyl phosphate synthetase. It activates it. يعني when we have N-acetyl glutamate we will activate carbamoyl phosphate synthetase 1 to start this reaction. حلو؟ بعدين the carbamoyl phosphate will combine with something called ornithine. Ornithine is a carrier, it's a transporter. It transports stuff, it takes stuff, puts them somewhere, then takes something else, puts them back. I'm going to show you how in a bit. For now, just know what I said. So carbamoyl phosphate will combine with ornithine, and through the enzyme ornithine transcarbamylase, we will get citrulline. All of this happens where? In the mitochondria. After we get citrulline, this is where we have to move to somewhere else. The citrulline will go to the cytoplasm. Hello? Citrulline will then combine with aspartate, and through the enzyme arginosuccinate synthetase, we will synthesize what من اسم الانzyme arginosuccinate. بعدين this arginosuccinate will break down into arginine and fumarate by arginosuccinase. We are cleaving it. The fumarate will go to the citric acid cycle and it will give oxaloacetate. So if you get a question that says, what is produced by arginosuccinate that will go to the citric acid cycle? You will be like fumarate. طيب, now let's go back to arginine and focus on it. Arginine will be again broken down into urea and ornithine. And this happens by the enzyme arginase. Do you guys remember how I said in ornithine is a transporter or it's a carrier? Why? Because after it gets خلينا نقول produced by arginine, it's gonna go all the way back to the mitochondria to combine with carbamoyl phosphate and do this reaction again. And then it will be produced by arginine and goes back. That's why ornithine is important, تمام? This is the urea cycle. This is the urea cycle. Memorize the enzymes I told you, the steps, what happens in the mitochondria, what happens in the cytoplasm. Or remember N-acetyl glutamate as an activator of carbamoyl phosphate synthetase 1. And remember fumarate as something that will go to the citric acid cycle. So urea synthesis takes place in the liver. طبعا something that's good to know is one nitrogen atom comes from aspartate. This is important to know. A question could be like, which amino acid gives us a nitrogen in urea cycle? You'll be like aspartate. Another nitrogen comes from ammonia, and a carbon comes from carbon dioxide. Remember this. As we said, ornithine acts as a carrier. Fine control of the urea cycle is through what? Regulation of carbamoyl phosphate synthetase 1, which is the rate limiting step. What regulates it? N-acetyl glutamate. This is the rate limiting step, ladies and gentlemen. Something I want you to know, it was asked about once, carbamoyl phosphate is the first donor of an amino group. Don't ask questions, just remember, wallahi, I saw it in a past question, carbamoyl phosphate is the first donor of an amino group. طيب, the concentration of N-acetyl glutamate, it's good to know, it's increased by what? By acetyl-CoA, glutamate, and arginine. طبعا you don't have to memorize this, but this is the sum of the urea cycle. You have the ammonia, the carbon dioxide, the ATP, and we will get urea. This is the most important. But now we're going to talk about the deficiencies, genetic deficiencies of the urea cycle. Two important enzymes اللي هم أول اثنين هذول carbamoyl phosphatase synthetase صح? What will happen if it's inhibited? Or ornithine transcarbamylase, what will happen if it's inhibited? تمام? If carbamoyl phosphate synthetase 1 is inhibited, it's deficient, this means we will have a lot of ammonia, hyperammonemia, elevated blood glutamine. Why? لأنه we're unable to do the urea cycle. فالأمونيا ammonia مو قادرين نحولوا اليوريا. It's gonna accumulate. حلو? But something to keep in mind. In this disease, orotic aciduria is absent. Why do I mention this? Because in ornithine transcarbamylase deficiency, which is the most common deficiency of the urea cycle, we will have orotic aciduria. We will have acid accumulation. طبعا in ornithine transcarbamylase deficiency, I want you to know that we will have the same symptoms: hyperammonemia, elevated blood glutamine, death, cerebral edema. The only difference is that we will have orotic aciduria in ornithine transcarbamylase. Something I want you to know that has been asked about before. حلو? Orotic aciduria بصفة عامة. What is it related to? Pyrimidine synthesis. That's all I want you to know. طبعا something I want you guys to know. If you get a question about urea cycle defect, but it doesn't mention orotic aciduria, what enzyme would you choose? You would choose ornithine transcarbamylase because it is the most common. تمام? Now let's go to the treatment. How do we treat a lot of, let's say, ammonia and all of these disorders? If we have glycine that's building up, we give benzoate. If glutamine is what's building up, we give phenylacetate. Don't ask questions, memorize these as they are. I've seen questions about them. خلاص, glycine, benzoate, glutamine, phenyl acetate. And now we finished the second lecture. Let's move on to the questions. Not the second lecture. Second lecture to me after my break. But in reality, this is lecture number 7, meaning we have 6 left, inshallah. So a 3-month-old child is being evaluated for vomiting. Lab shows hyperammonemia and what? Orotic aciduria. So what's the enzyme? Ornithine transcarbamylase. طيب, one nitrogen atom comes directly from ammonia. From which does the other nitrogen atom comes from? We said aspartate. طيب, this question, it's telling you this patient has hyperammonemia, increased blood glutamine, and orotic acid in the urine. What do we give this person? From the choices, what do you guys think is correct? Benzoate. طبعا benzoate احنا قلنا is for glycine, but here phenyl acetate is not an option. So just choose benzoate. But if phenyl acetate was an option, then you choose phenyl acetate because here they said glutamine. Okay, but this is correct. طيب, so a newborn with deficiency of mitochondrial carbonyl phosphate synthetase 1, which of the following changes is most likely the cause? Obviously, increased ammonia. Which of the following enzyme catalyzes the regulated step in the urea cycle? Yalla, you guys have to know this. Carbonyl phosphate synthetase 1. وترى carbonyl phosphate synthetase 2 is an actual enzyme. We will talk about it inshallah very soon. Very soon. طيب. Now خلصنا من what happens to nitrogen, how it's metabolized. I want to talk about amino acids degradation. How are amino acids broken down? First of all, I want to mention that the liver is the principal site of amino acid metabolism, except the branch chain amino acids. So all the amino acids are broken down in the liver, إلا the branch chain amino acids. Where are they broken down? In skeleton muscle. Remember this, they ask about it. So after removal of the amino group, the carbon skeleton, as we said, it undergoes further metabolism to be used as a fuel source. We already said this. طبعا, just as a reminder, do you guys know what the essential amino acids are? I think you do. If not, remember them. Private Tim Hall. I won't go through them, but I want you to remember them. Arginine usually is not 100% essential. Arginine, I want you guys to remember, it's conditionally essential. Meaning if مثلا we need it a lot, it becomes essential. But if we don't need it a lot, we already have enough of it, it doesn't become essential, we make it an hour. خلاص? But just know إنه it's conditionally essential. طبعا, inborn errors of amino acid metabolism, what are they? They are defects on the metabolic pathways of amino acids. And they will lead to a lot of issues, such as hypotonia or CNS issues. That's why newborn screening is important. We will talk about these defects. هذا كله كلام فاضي. Remember transaminases, because transaminases were mentioned once again in this lecture, but I won't repeat them, and it's exactly what I explained. Alanine aminotransferase, alanine and pyruvate, aspartate aminotransferase, aspartate, and oxaloacetate. Remember this like your names, خلاص? طيب. Now we're going to move on to a very important concept, ketogenic versus glucogenic. But I want you guys to understand why we're dealing with this. Now we have amino acids, صح? When they're broken down, the products of this breakdown are going to be used to do things, okay? The products will either give us ketones or glucose. But that's why some amino acids are ketogenic, meaning when they break down, they will become ketones, while others are glucogenic, meaning when they break down, they will become glucose. Or some are both. I want you to remember this Venn diagram, but there is an easy way to remember it. Only remember ketogenic will both. خلاص, everything else will be glucogenic. فالكيتوجينيك بس اثنين. They both start with L. Lysine ولوسين. هذول ketogenic. حلو؟ Both يعني can be ketogenic and glucogenic هم tryptophan, threonine, phenylalanine, tyrosine, and isoleucine. Everything else is what? Glucogenic. طبعا, something else I want you to know that can be asked about. Something that is ketogenic, what does it give? I just said it. Ketone bodies, keto acids, which are what? Be specific, acetyl-CoA and acetoacetyl-CoA. But what about something that's glucogenic? It gives glucose, but how? By giving pyruvate, oxaloacetate, fumarate, وهالأشياء that are in the glucose pathways. خلاص? I hope this concept is clear. طبعا, I want you guys to know there is an enzyme اسمه asparaginase. I don't want you to know anything about it except this slide. حلو? Asparaginase is an enzyme that converts asparagine into aspartate. من اسمها asparaginase. It will break down asparagine. So it will break down the asparagine into aspartate and ammonia. حلو? طيب. Why is this enzyme important? Because this enzyme, asparaginase, is used to treat acute lymphoblastic leukemia. You may be asking, how can we treat a whole cancer with this L-asparaginase? Here is the explanation. Normal cells, بالعادة, they synthesize asparagine. Leukemic cells, these cancer cells, they depend on asparagine to survive. So when we use asparaginase to break the asparagine into aspartate to ammonia, ما عاد حيصير في a lot of asparagine, meaning إيش؟ Meaning the cancer cells will die because there won't be asparagine for them, تمام? Moving to the last section of this lecture, the doctor had this slide. Well, the doctor talked about every single one of these pathways very briefly. And what I don't understand is why did the doctor include these pathways in this lecture? Guys, do you know in بعد lecture we're gonna have 3 other lectures. One of them will be a 2-on-1. The 3 next lectures will talk about these pathways in detail. So I don't get why the doctor had to include these pathways in this lecture specifically. But my job is to cover everything in every single lecture. So I'm going to talk about these pathways, but not in the full detail because this will be covered in the next 3 lectures. You'll have enough of it. I will talk about what was mentioned in this lecture in specific. تمام? First we have phenylalanine and tyrosine metabolism. What I want you to know is that phenylalanine can be converted to tyrosine. تمام? How? By an enzyme called phenylalanine hydroxylase. A phenylalanine hydroxylase turns phenylalanine into tyrosine. Something I want you to know is that phenylalanine hydroxylase, we will talk about this later. Guys, focus with me. Everything you remember now will help in the next 3 lectures. Wallah, focus with me. So phenylalanine hydroxylase needs a cofactor called BH4. خلاص؟ طبعا tyrosine بعدين it will give us a lot of things. It will give us epinephrine, norepinephrine, melanin, a lot of things. So thyroid hormones. خلاص? So that's the first section. Second, we have threonine, serine, and glycine metabolism. All I want you to know is that threonine can be converted to glycine. Hello? Why is glycine important? Because when glycine is converted into its later products, which is oxalate, this oxalate can cause kidney stones. فعشان كده we don't want to have too much glycine because too much glycine means too much oxalate, which means kidney stones. شايفين? Converted to oxalate, which can cause kidney stones. طيب, something I want you to know is that glycine cleavage, يعني when glycine is cleaved, which gives us مثلا serine or oxalate, it will give us a carbon unit that will be needed for tetrahydrofolate. Just know it as it is. خلاص? Tetrahydrofolate is an important vitamin or cofactor involved in this pathway. Then when glycine is cleaved, it will give us a carbon unit that is needed for tetrahydrofolate to work. Next section, methionine and cysteine metabolism. All I want you to know is that methionine will turn into something called S-adenosyl methionine, SAM. SAM is very important, by the way. We will talk about it later on. Oh, please remember this tiny box. Wallahi, it's asked about. The question would be like, what is a major methyl group donor? The answer is SAM. Donates methyls. تمام? طيب. SAM will later become homocysteine at the end. طيب. Homocysteine. What will happen to homocysteine? There are 2 fates of homocysteine. Either it becomes cysteine by transsulfuration, or homocysteine can go back all the way to methionine. How? By a process called remethylation. فالهوموسيستين يا حيكمل ويصير cysteine, this would require vitamin B6, or the homocysteine will go back to become methionine, will start the reaction again, this would require B12. And this process is called remethylation. خلاص? But this is exactly what we said here. طبعا folate is needed. I just want to mention إنه folate is needed for these reactions اللي هنا for remethylation specifically. If we want homocysteine to go back to methionine, it needs vitamin B12 and folate. What kind of folate specifically? N5-methyl-THF. خلاص, it's good to know it. طيب, then we have histidine metabolism. Histidine, through histidase, will become urocanic acid. Don't want you to know this. What I want you to know, which is very important, is figno, N-formimino-glutamate. Why is it important? Because it's converted to glutamate. It becomes glutamate. We know how glutamate is important. But to be converted to glutamate, it needs tetrahydrofolate. The tetrahydrofolate is what converts the figlu to glutamate. If we don't have tetrahydrofolate, we can't make glutamate from figlu, so figlu will accumulate. So the excretion test of figlu is an important marker for folate deficiency. That's why figlu is important. If someone has high figlu, we would know high figlo, a lot of it being excreted in the urine, معناتها it's building up. That's how we know folate is deficient. Next, we have branched chain amino acid metabolism. All I need you to know is in valine, isoleucine, and leucine, other than the fact that they are metabolized in skeletal muscle, they have a special enzyme for them. What is the enzyme called? It's called branched chain alpha keto acid dehydrogenase, BCKD. Something interesting about this enzyme is that it doesn't just need one cofactor or two cofactors, it needs five cofactors. Vitamin B1, TBB, lipoic acid, vitamin B5, which is CoA, vitamin B2, which is FAD, and vitamin B3, which is NAD+. You have to remember this, guys. Wallahi, you could get an SAQ. I don't know if you have SAQs, but it could be list the cofactors required for BCKD. You need all of these cofactors. Memorize them, remember them. But later on, we will talk about the disease where this enzyme is deficient, meaning we will have a buildup of branched chain amino acid. This disease is maple syrup urine disease, where there will be a distinct maple syrup odor to the urine and breath. Tamam? Propionyl-CoA pathway. It was covered in this lecture, but guys, صراحة, I will not talk about it because I already explained it in detail with fatty acid vectors. It's the same. Propionyl-CoA carboxylase biotin, methylmalonyl-CoA mutase, vitamin B12. I already talked about this in zaman, so just review it for your own sake. And this lecture is over. Let's go to the questions. طيب, asparaginase can be used as a chemotherapy. What reaction is catalyzed by asparaginase? I told you, asparagine to aspartate. طيب, in the propionyl-CoA pathway, which amino acid does not contribute to the pathway? Do you guys remember when I told you that there are 4 certain amino acids that go to the propionyl-CoA pathway? If not, then you go back here and remember. Propionyl-CoA pathway, only 4 amino acids go into this pathway: valine, isoleucine, methionine, and threonine. But which one isn't included? Leucine. This question, an amino acid is called ketogenic if its catabolism leads to which of the following? Acetyl-CoA. Which of the following is both glucogenic and ketogenic? هذه من Venn diagram. Easy questions. طيب. Guys, here I don't want to know anything. I just want to know oxalate. Oxalate is associated with what? Glycine. خلاص? طيب, now we're going to talk about the metabolism of phenylalanine and tyrosine. المشكلة in the table قبل شوي, we just talked about it, but I guess now we have to go into more detail. Very easy, by the way. The first step is what I already told you guys من زمان. إنه phenylalanine becomes tyrosine. How? By phenylalanine hydroxylase. And this enzyme requires what? BH4. طيب, let's say that phenylalanine hydroxylase is deficient. What disease will we have? PKU, phenylketonuria. We will have accumulation of phenylin because we don't have the enzyme that will make it tyrosine. خلاص? طيب, in a normal case, what will happen to tyrosine? طبعا before I speak about that, sorry, I want to talk about BH4. أنا قبل شوي I simply told you خلاص guys, phenylalanine hydroxylase needs BH4. But what I want you to know about BH4, الحمد لله back to normal. BH4 is called tetrahydrobiopterin. This is its full name, and it needs NADH. But this is all I want you to know. And throughout this reaction, the BH4 will become BH2. The BH4 is called tetrahydrobiopterin, and it needs NADH. طيب, how does the BH4 become BH2? Through the enzyme dihydropteridine reductase. خلاص? طيب, let's go back to the tyrosine. شايفين ليش في 3 arrows? Because there are 3 different pathways. Tyrosine can become a lot of different things. One of them is the pathway of catecholamines, epinephrine, norepinephrine, وكذا. How does this happen? First of all, tyrosine is converted to dopa by the enzyme tyrosine hydroxylase. This requires BH4. Dopa من اسمها dopa, it will become dopamine. This happens by the enzyme dopa decarboxylase. I want you to remember these 2 points. And dopa decarboxylase is activated by vitamin B6 or inhibited by carbidopa. خلاص, remember this. Then dopamine will become norepinephrine by dopamine beta hydroxylase, and this enzyme requires vitamin C. شايفين guys, every single enzyme requires something, and you have to memorize it, okay? Yeah, I can't explain way too much here. It's genuinely memorization. بعدين we have the norepinephrine. What will it become logically? Epinephrine. How? By an enzyme called phenyl phenylethanolamine and methyltransferase, doesn't matter. What I want you to know is that this reaction, norepinephrine to epinephrine, involves donating a methyl group. ومين قلنا is the person that donates methyl? Sam. خلاص, is that clear? طيب, let's move on to tyrosine, the other pathway of tyrosine. Or actually, before that, I actually want to talk about SAM. SAM, we talked about it قبل شوي, I will talk about it again. But we said that SAM will become homocysteine. Homocysteine, if you guys remember, we said that it has two pathways. Either the homocysteine becomes cysteine, which would need B6, or it would go back to become methionine and SAM again. How? By B12 and folate. خلاص? طيب, tyrosine can also become thyroxine. Tyrosine, thyroxine, they rhyme. How does this happen? We need iodine. That's all I want you to know. Last but not least, tyrosine through the enzyme tyrosinase can become melanin. You know, melanin pigment gives the dark pigmentation of the skin. طيب, if someone has a tyrosinase deficiency, it means that the tyrosine can't become melanin. So this person will not have melanin. What does this mean? It means that this patient will have a disease called oculocutaneous albinism. تمام? I don't know why this is very annoying. Whatever. Okay. طيب, phenylketonuria. We just talked about it. We have an absence of the enzyme phenylalanine hydroxylase, which is the most common cause. The most common cause in this enzyme is deficient, phenylalanine hydroxylase, or BH4 is deficient. لأنه even if we have phenylalanine hydroxylase, if there isn't the BH4 necessary, إيش استفدنا? You know? طيب. So these are the causes of phenylketonuria. What will happen? I already told you, accumulation of phenylalanine and deficiency of what? Tyrosine. Signs are musty smell in breath, not maple syrup smell and maple syrup urine disease. That musty smell, it doesn't smell good. In breath, sweat, and urine. And we will also have CNS symptoms and pale skin with blue eyes. ليش blue eyes? لأنه we don't have melanin. Tyrosine will not give us anything. فحتى الثايرويد حيكون نازل. طيب, how is it treated? Treated by giving tyrosine and avoiding phenylalanine. But something also important that I want you to avoid in a patient with PKU is what? Aspartame. Why? Aspartame, what is it? It's a sweetener that has phenylalanine. We don't want to give phenylalanine, we want to avoid it. فإن a patient with PKU, what do we avoid? Aspartame. Remember this, very important. طيب, Parkinson's disease. Parkinson's disease happens when we don't have a lot of dopamine. How do we treat it? We give L-DOPA because DOPA gets converted into dopamine. But there is an imposter, carbidopa. Why do we give carbidopa? إذا الدوبامين عندنا قليل. Why do we give carbidopa, which literally inhibits the formation of dopamine from dope? Here is the reason. The reason is, guys, L-DOPA can be converted in the brain where we need it and in the body, the circulation. We don't want it to be converted in the circulation. ما نبغى الدوبامين يكون بالcirculation. We want it in the brain. So we give carbidopa because carbidopa will inhibit L-DOPA from becoming dopamine. But here's the catch. Carbidopa does not cross the blood-brain barrier. So L-DOPA will be inhibited by carbidopa until it gets to the brain. But we wouldn't have wasted any of the dopamine على circulation. And now the L-DOPA alone will be in the brain and it will give us dopamine. طيب, now oculocutaneous albinism. I already talked to you guys about it. Decreased melanin. It can either, and obviously the signs are pale skin, blonde hair, blue eyes, could lead to skin cancer, sunburns. Well, there are two subtypes. Oculocutaneous albinism type 1, which is tyrosinase deficiency, or type 2, which is by a tyrosine transporter's defect. تمام? طيب, now we want to talk about how tyrosine is degraded. يعني خلاص, we have tyrosine. Okay, it became transformed into different stuff, but how do we break it down and get rid of it? طيب, tyrosine will become something called homogentic acid. Well, this homogentic acid has to be broken down into fumarate and acetoacetate. By what enzyme? Homogentic acid oxidase. If homogentic acid oxidase is deficient, what do you guys think will happen? We will have buildup of homogentic acid, which would lead to a disease called alkaptonuria. Very special and interesting disease we will talk about now. طبعا, this is for tyrosine. But what about epinephrine, norepinephrine, dopamine? How are they broken down? Very easy. Two enzymes that break down all of these catecholamines. monoamine oxidase and catechol methyltransferase اللي هم ال MAO and COMT. حلو. Epinephrine and norepinephrine will be broken down by these enzymes into something called metanephrines. Then this metanephrine will become vanilmandylic acid VMA بعدين يروح على اليورين. Dopamine will be broken down into not vanilmandylic acid into homovanillic acid HVA, and then it goes into the urine. But what I want you to know from here, monoamine oxidase, catechol methyltransferase, they break down catecholamines. Catecholamines, epinephrine, norepinephrine will become metanephrines, then VMA, while dopamine becomes HVA. خلاص? طيب, let's talk about alkaptonuria. We already did, but in more detail. Deficiency of what? Homogentic acid oxidase. So we will have buildup of what? As we said, homogentic acid. For what will this tyrosine breakdown? We don't have homogentic acid oxidase. These are the pictures in your slide that can come in your exam. In cartilage and joints. طيب, let's talk about pheochromocytoma. Very big. It will cause symptoms of high epinephrine, headache, palpitation, sweating, وهكذا. Very simple. What is the best test for pheochromocytoma and catecholamines? You have to remember this. Related to pheochromocytoma بس حطيته هنا. Monoamine oxidase inhibitors. What happens if we inhibit monoamine oxidase? We won't break down dopamine with catecholamines. The monoamine oxidase inhibitors are used to treat depression because they won't break down the dopamine for the dopamine. That's clear on why we use monoamine oxidase inhibitors. That lecture is over. Does it donate? We already said methyl group. VMA is a metabolite of what? Epinephrine. This picture, which clearly shows black pigmentation in joints. What does it show? Toxic substance accumulating due to a defect in tyrosine metabolism. طيب. Hypopigmented irises, translucent skin, light hair. Come on. This is a amino acid. We know, tyrosine. طيب. 3-year-old girl born at home, local clinic because of seizures. She has retardation, musty body odor. pH4. So what is the correct measure? Oh, what is the disease? PKU. But what is the measure that we should take? Low phenylalanine. Tamam? I'll take another short break, a final one, and then we'll continue the final lectures of this PAL, which are 4 lectures, but this one is a 2-in-1, so باقي لنا بس 3 lectures, inshallah. I'll see you after a short break. Let's continue with the final portion of this PAL session, inshallah. We're going to cover 4 more lectures. As I said, the first 2 are 2-in-1. I did them as a 2-in-1 اللي هم هذول الاثنين. Then Purine Metabolism and Pyrimidine Metabolism. So let's get into this lecture. It's literally everything that I explained to you in that one lecture where there was the table and I was like, why did the doctor include them now? These are the exact same things. But now we're going to go into a bit more detail. وفي a bit of news. خلاص? بس everything is going to be easy inshallah. طيب, let's begin. First thing I want to talk about tryptophan. You guys already know in heart lung disease, tryptophan is not converted into vitamin B3 because tryptophan is not being absorbed. But you know this. You know in that tryptophan gives us vitamin B3. How? By the enzyme tryptophan dioxygenase. And for tryptophan to make vitamin B3, it needs vitamin B6. خلاص, I need you to know this. طيب, what is the new pathway that you guys don't know yet about tryptophan? Is that tryptophan, through tryptophan hydroxylase, can become 5-hydroxytryptophan. Tryptophan hydroxylase, this first steps needs BH4. خلاص انتوا تعودتوا على BH4. طيب, why is this significant? Because this 5-hydroxytryptophan can become serotonin, a very important neurotransmitter. Okay? طيب, how does 5-hydroxytryptophan become serotonin? By aromatic L-amine acid decarboxylase. You don't have to know the enzyme. Just know that it requires vitamin B6. وإن Tryptophan gives us serotonin. Last but not least, serotonin will also give us melatonin, but it requires acetyl-CoA to do that. Hello? But tryptophan is important. It gives us vitamin B3. It gives us serotonin. It gives us melatonin. You guys need to know this. خلاص? Remember the enzymes. Remember what each enzyme requires. طيب. As I said, guys, already, defective tryptophan absorption in heart nerve disease, we already discussed this, will lead to vitamin B3 deficiency. So pellagra-like syndrome, dementia, diarrhea, and enteritis. In this section, we already talked about it. Histidine to histamine by histidine decarboxylase. Hello? طبعا histamine is important in inflammation. That's why the amino acid histidine is important because it can take us to histamine. And it requires pyridoxine, which is basically vitamin B6. طيب, glycine, we also talked about glycine already, how it gives us oxalate, which can cause kidney stones. Something else I want you to know about glycine, and it forms the structure that is found in the RBC heme ring. You guys know an RBC has hemoglobin and hemoglobin has a ring. طيب, who creates this ring? Glycine. Glycine and succinyl-CoA will combine and turn into ALA. This ALA will form the porphyrin ring in the heme. خلاص? This is very important. So the porphyrin ring in heme is ALA, which is formed from glycine. But what's the enzyme used to make ALA? ALA synthase. Well, again, it needs pyridoxine, which is vitamin B6. By the way, this is like 2 lectures, 60 slides. This table summarizes all of it, I promise. Then we have arginine. Arginine has a bunch of fates. Hello? You guys already know one of them. Arginine through arginase becomes urea or lithium. In the urea cycle, you guys know this. Something else that I want you to remember زي اسمكم هو إنه arginine becomes nitric oxide. Don't ask questions. خلاص? Arginine gives nitric oxide. You might get a question. Nitric oxide, the vasodilator, what amino acid produces it? The answer would be arginine. Now let's talk about the creatine pathway. You don't have to know all of these details. All I want you to know is that arginine, it can combine with glycine and SAM, I want you to know SAM, to give us creatine. Phosphocreatine is basically the reservoir of creatine, and it keeps energy in the muscle. That's the function of creatine. طيب, how do we get rid of creatine? By changing it into creatinine. خلاص? That's all I want you to know from this section. And then we have glutamate. Glutamate, guys, بعادته, it's an excitatory neurotransmitter. It causes contractions, excitation, but it can be converted to GABA, which is an inhibitory neurotransmitter. GABA is literally just the opposite of glutamate, guys. عرفتوا? طيب, what enzyme does this? Glutamate decarboxylase. Needs vitamin B6. خلاص? Glutamate excitatory, GABA inhibitory, glutamate decarboxylase to switch between them, and the vitamin needed for this enzyme is B6. Next, branch chain amino acids. We already talked about this, guys. We literally did valine, isoleucine, leucine. How are they degraded? أول شيء where? In skeletal muscles. How? By the enzyme. Branch chain, alpha keto acid dehydrogenase, BCKD. And remember, what were the 5 cofactors it needed? TPP, which is B1, lipoic acid, CoA, which is B5, FAD, B2, and NAD, B3. We already went through this. Well, I told you guys, and if this enzyme is deficient, we will have maple syrup urine syndrome. We will talk about it in this lecture. طيب, methionine homocysteine metabolism. We literally talked about this too. إنه methionine will become SAM. SAM will become homocysteine. Homocysteine has 2 choices, either to become cysteine or to become methionine. How does it become methionine? By methionine synthase. Well, that would require folate and vitamin B12. خلاص? Now I'm feeding you a bit more details. There are enzymes involved. Methionine synthase requires folate and vitamin B12 to turn homocysteine into methionine. طيب, what about homocysteine when it wants to get to cysteine? How does it become cysteine? First, it becomes cystathionine. Cystathionine, sorry. Cystathionine. So homocysteine, cystathionine, then cysteine. طيب, how does homocysteine become cystathionine? By an enzyme called cystathionine synthase. من اسم الانزيم. Well, this enzyme requires vitamin B6. طيب, what if this enzyme, cystathionine synthase, is deficient? What condition will we have? We will have homocystinuria, buildup of homocysteine. And this homocysteine will not be able to be converted into cystathionine and cysteine. So it will build up the homocysteine, so we will have homocystinuria. Then the cystathionine will be converted into cysteine by an enzyme called cystathionine lyase, which also requires vitamin B6. خلاص? طيب, but obviously deficiencies in vitamin B12 and folate will prevent homocysteine from going back to methionine, and deficiencies in cystathionine synthase will prevent homocysteine from becoming cystathionine, which is the classic homocystinuria. خلاص؟ طيب, last is for the 50 millionth time البروبيونيل-CoA pathway, valine, isoleucine, methionine, threonine. I won't go through it, guys. We went through it 70 مرة. It's the same exact thing, same enzymes, which one needs biotin, which one's B12. حتى the diseases we're going to talk about اللي هم propionic acidemia, it just means إنه propionyl-CoA is accumulating لأنه we don't have propionyl-CoA carboxylase or we don't have biotin. وmethylmalonyl-CoA acidemia means إنه we don't have methylmalonyl-CoA or we don't have B12, which you know is needed. Methylmalonyl-CoA mutase, sorry. So methylmalonyl-CoA will accumulate. Simple as that. طيب, let's talk about maple syrup urine disease. It's caused by deficiency of what? As we said, branch chain alpha keto dehydrogenase, which will lead to an accumulation of branch chain amino acid. طيب, what is the characteristic of this disease? I said it before. Sweet maple syrup-like odor of urine. خلاص? طيب, we said in the branch chain amino acids are valine, isoleucine, or leucine. طب, which one of them causes the maple syrup-like odor? It's isoleucine. طب, which of the three branch chain amino acids causes neurotoxicity? Leucine. خلاص. طبعا the treatment of maple syrup urine disease, a lot of treatments, but one of them thiamine supplementation, B1. Why? Because if you remember, لو كنتم مركزين, the enzyme needs 5 cofactors. One of them was what? B1. So we give B1 to potentially help the enzyme work. خلاص. طيب, now we're going to talk about homocystinuria. We already said إنه it's caused by what? Cystathionine synthase deficiency. It could be also a deficiency of what we already said, methionine synthase, folate, B12, all of them will cause homocystinuria. But classic homocystinuria, most commonly, it is caused by cystathionine synthase deficiency. خلاص? طيب, what are the signs of homocystinuria? يعني you have a patient in front of you. How can you know they have homocystinuria? Lens dislocation, long limbs, and chest deformities. Chest deformities that look like this. تمام. طبعا dietary treatment is how? Avoid methionine and take cysteine and B6. That's all you need to know. طيب, propionic acidemia and methylmalonic acidemia, we already talked about it. I won't even go through it again. Just know when it causes neonatal metabolic acidosis because we have acidemia, logically. Yeah. And these 2 lectures are over. طيب, والله من جد, you guys don't need to know anything else. My advice for you is go to this table. طبعا credits والله to ChatGPT. You guys will understand all of these graphs. تعبت مرة والله مع ChatGPT to make them. I didn't get them from textbooks or your slides. I made them myself by the help of ChatGPT, where they literally include all the information you need. So when you guys revise or review my slides or review my pad session, after watching it the first time, genuinely just depends on these graphs. عرفتوا? طيب, questions. High homocysteine level due to deficiency of what? B12. Correct neurotransmitter with its precursor, melatonin, and tryptophan. Long limbs, chest deformities, come on, classic homocystinuria, elevated homocysteine levels, deficiency in which enzyme? Cystathionine synthase. طيب, which amino acid is involved in formation of both heme and creatinine? We know it, glycine. قلت لكم, glycine does the heme through ALA, and glycine combines with aspartate to make creatine. But aspartate doesn't make heme. The question wants both. Which amino acid does both the heme and the creatine? طيب, urine has a characteristic odor of maple syrup. Well, guys, by the way, I'm sorry if I'm going through the questions fast. But again, there's no one to interact with me. But I want you guys to pause the questions, answer them yourself, then see me answer them. This is maple syrup urine disease. This is the enzyme. طيب. Ectopic lens. Developing a clot. What is this? Cystathionine synthase. Homocystinuria. طيب. This question. It's about methylmalonic acidemia. Results from a defect in which enzyme? Methylmalonyl-CoA mutase. طيب, disorder affecting branched chain amino acid. Which enzyme requires, the enzyme requires which cofactor? We know it. B1, B2, B3, B5, lipoic acid. One of them, which happens to be in the choices, is thiamine. That's why, guys, in maple syrup urine disease, we give thiamine as a treatment. We already said that. طيب. Now the final two lectures, purine metabolism and pyrimidine metabolism. Let's start by setting foundations. Hello? If you guys remember from my past session in the beginning of the year, I told you the nucleotides are a base, a sugar, and a phosphate. This is how nucleotides are. Hello? This is what they're made. The purine bases هم pure as gold. عندنا adenine وguanine. You guys know this. There is a new one that we will talk about is inosine. Inosine is basically the baby form. It will then turn into adenine and guanine. Tamam? Just keep that in mind. What are sources of nucleotides, the purines especially, either from the diet or endogenous, yani from the body, not from outside. From the body, we can either create them or salvage them. What does salvage them mean? Yani we get a guanine, we break it down, and we take the nucleotide from it. عرفتوا? طيب. I want you guys to know the precursor molecules for purine synthesis. They're important, especially glutamine, glycine, aspartate, carbon dioxide, and tetrahydrofluorate. Very important. I want you to remember them. خلاص? طيب. Now we're going to start by de novo synthesis of purine nucleotides. In purine nucleotide synthesis, we start by creating the sugar, the ribose sugar, and then we add the base. حلو? In pyrimidine بالعكس, we do the base, then the sugar. فlet's do the sugar مع بعض. يلا. We start with ribose 5-phosphate. Ribose 5-phosphate through the enzyme PRPP synthetase will give us PRPP, which is 5-phosphoribosyl 1-pyrophosphate. Very important. And the enzyme PRPP synthetase, I want you to know that it's regulated by what? It's activated by phosphate and inhibited by the purine ribonucleotides, which are the end products. Logical. طيب بعدين let's go to the rate limiting step, the regulated step, which is PRPP turning into the final sugar we want, which is 5-phosphoribosylamine. How does this happen? By the enzyme glutamine phosphoribosyl pyrophosphate amidotransferase. You don't have to know the full name. I just want you to remember that it's an amidotransferase. And this amidotransferase is the committed and regulated step. What is it inhibited by? It's inhibited by AMP and GMP اللي هم adenosine monophosphate and guanosine monophosphate, which are the end product. خلاص؟ طبعا something I want you to know is إنه obviously tetrahydrofolate is needed in the purine ring. But why am I putting it here, mentioning it here? Because methotrexate and sulfonamides are drugs that inhibit tetrahydrofolate. What will this lead to? It will lead to the inhibition of synthesizing nucleotides. Keep this in mind for later on. خلاص? But this is us making the sugar, ribose 5-phosphate, to PRPP by PRPP synthetase activated by phosphate and inhibited by purine ribonucleotides. Then this PRPP through the amidotransferase, which is the regulated step, will become phosphoribosylamine, which is the final sugar we need, inhibited by AMP and GMP and activated by PRP. طبيعي. So the reaction goes that way. خلاص? Now, again, the purine ring composition. We already said this in the previous slide. Glycine, aspartate, glutamine, CO2, and tetrahydrofolate. خلاص? طيب. Now let's go to the nucleotide itself, اللي هو contains the base. The first nucleotide created, just as I said in the previous slide, which is the baby, the first one created in purine metabolism is inosine monophosphate. And what is the base of it? Hypoxanthine. Then this IMP will change into either adenine monophosphate or guanine monophosphate through different enzymes. The most important enzyme is IMP dehydrogenase. IMP dehydrogenase converts IMP into GMP. وكمان AMP, by the way. IMP dehydrogenase can make it IMP. Something I want you to know. Mycophenolic acid. This is an acid that inhibits IMP dehydrogenase. What will this lead to? We won't get GMP and AMP. Just keep this in mind. Now, last but not least, I want you to know in the nucleotides that are required for DNA synthesis are deoxyribonucleotides. But see, it's a ribose sugar, not a deoxyribose. So how do we change it from ribose to deoxyribose? We get the ribonucleotide, And through the enzyme ribonucleotide reductase, we get deoxyribonucleotides. خلاص? Activated by ATP and inhibited by deoxy ATP. خلاص? This is what I want you to know. Ribonucleotide reductase gets the ribonucleotide and makes it as a deoxyribonucleotide. Now take a look at this beautiful graph. This is the whole lecture. Methotrexate, sulfonamide, and mycophenolate in more detail. Methotrexate, what is it, guys? It's a structural analog of folic acid. It looks like folic acid. It inhibits the enzyme dihydrofolate reductase. تمام? When do we use methotrexate in cancer treatment? An adverse effect of it is bone marrow suppression. لأنه we are inhibiting the formation of DNA and nucleotides. We have bone marrow suppression. We're unable to create new cells. خلاص? Just remember, methotrexate inhibits dihydrofolate reductase and it's used in cancer treatment. Then we have sulfonamides. Sulfonamides and trimethoprim, they are similar to methotrexate. They are involved in the folate pathway, but there's a difference. They are antibacterial and they don't inhibit dihydrofolate reductase. They inhibit bacterial folate synthesis. They inhibit the synthesis of folate in the first place. طب why is this significant? Because إحنا humans, we don't even synthesize folates. So when we take these antibiotics, they don't affect us. Our folate will stay the same. It will only affect the bacteria. خلاص? Remember this about sulfonamides and trimethoprim. Last but not least, mycophenolate. Mycophenolate, قلنا, it will inhibit the enzyme IMP dehydrogenase and inosine monophosphate. Inosine monophosphate, yeah. It will stop the formation of adenosine monophosphate and guanosine monophosphate. So this is the de novo pathway. This is how we create purine nucleotides from scratch. تمام? Now we're going to go to the salvage pathway, which is basically recycling. طيب, the purine salvage pathway basically means that we are recovering the bases instead of creating them من جديد. How? By phosphorybosylation. طبعا the sugar is always going to be the PRPP. To recover the bases, what do? We take adenine and we convert it back to AMP through the help of PRPP. This is done by an enzyme called adenine phosphoribosyl transferase, APRT. So we are recycling the base, adenosine monophosphate, but not by creating them from inosine. لا, we are just breaking down adenine through this enzyme APRT and through PRPP and we get AMP. خلاص? Another one which is much more important صراحة is the hypoxanthine guanine phosphoribosyl transferase, HGPRT. This enzyme, it's similar to APRT, but because it's H and G, it will act on hypoxanthine and guanine. Breaks the hypoxanthine, gives us IMP, breaks the guanine, gives us GMP. Hello? Something important I want you to know. When we have a deficiency of HGPRT, we will have a syndrome called Lesch-Nyhan syndrome. This will lead to an inability to salvage IMP with GMP. We're going to talk about this in a minute. Just remember Lesch-Nyhan syndrome. تمام? طيب, adenosine deaminase deficiency. I'm going to first introduce what this enzyme is. Hello? Purines are generally أصلا converted to uric acid and excreted in urine. Who does this? Adenosine deaminase. Adenosine deaminase takes the purine, makes it uric acid, ويلا مع السلامة. طيب, when this enzyme is defective, we will have accumulation of adenosine, and no one is breaking it down. But the most important thing that will accumulate is what? DATP, deoxyadenosine triphosphate. Why is this important? Because when we have too much of D, it means we will inhibit ribonucleotide reductase. نرجع هنا. We will inhibit ribonucleotide reductase because we have a lot of D, the DATP. But we inhibit the ribonucleotide reductase, but we will not have DNA synthesis. Then we don't have deoxyribonucleotides anymore. And what will this lead to? A condition called severe combined immunodeficiency, SCID, which will cause a decrease in T and B cells. Very important. Please know it. خلاص? Adenosine deaminase, which usually breaks down purines, when it's deficient, we will have accumulation of DATP. Too much DATP inhibits ribonucleotide reductase. So the ribonucleotides will no longer be turned into deoxyribonucleotides. فthis will lead to no DNA synthesis, and it will lead to a condition called severe combined immunodeficiency, SCID. Memorize this very well. Next condition is gout, which is hyperuricemia. بس أول شيء, what is gout? It is deposition of urate crystals in joint, monosodium urate crystals. حلو. How do patients of gout present? As acute attacks of redness, pain, and swelling. They would come to you in a joint in their body, most commonly the big toe, which will be painful, red, and swollen. خلاص؟ طيب, how can we diagnose gout and confirm it? By identifying needle-shaped monosodium urate crystals under polarized light. Memorize this like your name. خلاص? This is the diagnosis of gout and how it's diagnosed. طيب, how do we treat gout? A drug called allopurinol. حلو. طبعا أول شيء I want to explain to you why it happens in the first place. مو إحنا قلنا إنه purines they get degraded into uric acid. طيب, when we have so many purines, they're gonna get degraded into so many uric acid. Well, this uric acid will be precipitated in the joints, which will cause gout. حلو. فallopurinol, what will it do? It will inhibit the production of uric acid by inhibiting xanthine oxidase. احنا بالعادة we have xanthine, it is broken down to uric acid by xanthine oxidase. Allopurinol will be like, no, we are not breaking down the purines anymore. We are not making any more uric acid from xanthine. So allopurinol will block xanthine oxidase, thereby reducing uric acid production. I want you guys to know something important. These are considered risk factors in people who can develop gout. Seafood or meat has a lot of purines. A person that eats a lot of seafood can get gout because he's getting a lot of purines. Similarly, a patient or a person that's spending his time in low temperatures might get gout. You want to know why? In the low temperatures will reduce the solubility of the urate. It will precipitate as crystals in the joint. خلاص, memorize these risk factors, very important. This is redness, swelling, and pain of a joint, and this is the needles of monosodium urate crystals seen under polarized lights. Right. Next syndrome. Next syndrome is the tumor lysis syndrome. تمام. What is tumor lysis syndrome? It is a complication of cancer treatment. Cancer treatment is chemotherapy. We are killing the cancer cells. المشكلة, when we kill too many of these cancer cells, we will get too many death of the cancer cells. These cancer cells will release the nucleic acids in them. For the nucleic acids will become purines, then to uric acid. But too many cells are dying, which means too many nucleic acids are being thrown out or exploded because the cells are dying. These nucleic acids will become uric acid. So we will get hyperuricemia. خلاص, this is tumor lysis syndrome. Last but not least, Leishman syndrome. We said deficiency of HGPRT, which is required for the purine salvage pathway. But what will happen? Loss of HGPRT will lead to reduced purine salvage. يعني we will have increased PRP or increased purine degradation or less recovery of the purines. But this will lead to hyperuricemia. And now we can't have a salvage pathway. We can't recover the nucleotides, for they're being degraded. So we will get hyperuricemia. What I want you to know about Leishman syndrome, very important characteristic feature, is it involves the biting of the lips and fingers. If you see a scenario where a child has self-injurious behavior such as biting lips or fingers, Leishman syndrome. على طول. Okay? طيب, this is a summary slide I made for you guys. I'm not gonna talk about it لأنه I said everything already. I just did it for you guys to review all the syndromes at once. So حرفيا it includes everything you need, by the way. طيب, checkpoint. We have a few questions. Biting fingers, HGPRT. على طول. Someone who has a lot of infections means إيش معناتها؟ إنه the B cells and T cells are not working well. What deficiency causes decreased B T cells because of decreased DNA synthesis? Adenosine deaminase. طيب, first fully formed purine nucleotide in purine synthesis, we said it, IMP. طيب, this is a scenario about gout, and we have a lot of purine degradation. That's how gout happens. You take a lot of seafood, a lot of purine is becoming uric acid. طيب, gout attack, allopurinol. What does allopurinol do? It inhibits xanthine to uric acid. How? By inhibiting the enzyme xanthine oxidase. خلاص? Last lecture. يلا هانت. Pyrimidine metabolism. بيراميدينز ايش هم؟ ثايمين وسيتوزين ويوريسيل. ثايمين in DNA and uracil in RNA. طبعا in purine احنا قلنا we make the sugar phase first then the base. In pyrimidine metabolism or synthesis we make the base first then we attach it to the PRPP. خلاص. I want you to know the precursors of pyrimidine synthesis. Aspartic acid, glutamine, carbon dioxide, and tetrahydrofolate. Very important to know. The product, the base that we will make is called uridine monophosphate. Very important to know. طيب, with a regulated step in this reaction of pyrimidine synthesis is carbamyl phosphate synthetase 2. We will talk about these enzymes right now. So pyrimidine synthesis. We start with glutamine. Glutamine is the starting point. That's why I mentioned it here, glutamine. It's a precursor. So we start with glutamine. Glutamine through the enzyme carbamyl phosphate synthetase 2, which is the regulated rate-limiting step of pyrimidine synthesis, will become carbamyl phosphate. Does this ring a bell, والله guys, صراحة it's too hectic. 3 hours بديت ألخبط. Does this ring a bell? The bell that should be rung by this is إنه carbamyl phosphate is involved in the urea cycle. It's involved in what the urea cycle. احنا قلنا in the urea cycle we start with ammonia through carbamyl phosphate synthetase 1 we get carbamyl phosphate. But here it's different. شفتوا هذا X. This is a different enzyme. Carbamyl phosphate synthetase 2. It takes glutamine and it changes it to carbamyl phosphate. تمام? Carbamyl phosphate synthetase 2, which is the regulated rate limiting step, is activated by ATP and inhibited by what? Logically, uridine monophosphate, since it's the end product. This carbamyl phosphate through aspartate will give us orotic acid. Very important. Keep it in mind. بعدين, this orotic acid through the enzyme UMP synthase will finally give us what we want, اللي هو uridine monophosphate. طبعا the more PRPP we have, the more UMP synthase will work. So PRPP will activate UMP synthase. طيب, let's talk about this tiny section here. Imagine if we have a deficiency in UMP synthase. What will happen? It means we can't turn orotic acid into uridine monophosphate. So what will build up? Orotic acid. So we will have orotic aciduria. خلاص? طيب, in normal cases, the UMP will then be converted into CMP, cytosine, and TMP, thymine. But something I want you guys to know. Thymine is obviously only in DNA, while uracil is only in RNA. طيب, if we have a deoxyuridine monophosphate, how do we change it into thymine? Then we can't use DUMP in DNA because it's uracil. It has to be thymine. Even if you forgot everything I just said, just remember this one thing. The enzyme thymidylate synthase will change the DUMP into DTMP. As simple as that. طيب, thymidylate synthase is inhibited by a certain medication اسمه fluorouracil, 5-fluorouracil. خلاص? 5-fluorouracil inhibits thymidylate synthase, which means we won't have DUMP becoming DTMP. Something else I want you to know is that methotrexate actually affects this reaction. Why? Methotrexate, what does it inhibit? Folate. This reaction, thymidylate synthase, this enzyme, it needs folate. So when we use methotrexate, we are also inhibiting this reaction. خلاص? Now let's go to orotic aciduria. We said it comes from a UMP synthase deficiency, which would lead to buildup of orotic acid and loss of pyrimidines, and we can't synthesize them anymore. What are the key findings? 2. Orotic acid in urine and megaloblastic anemia. There's something important about the megaloblastic anemia. It is not curable by vitamin 12. Yeah, if we give vitamin B12, the orotic acid will not be cured. The megaloblastic anemia will not be cured, even after we give vitamin B12. How is it treated? By giving uridine. This came in a question once. Please memorize it. Orotic aciduria, it means we can't create uridine ourselves, so we just give the patient the uridine. طبعا this is megaloblastic anemia. Something I want you guys to know. Do you guys remember in the urea cycle كان عندنا ornithine transcarbamylase deficiency? I'm gonna go back to it actually and use it as a chance to breathe. Here. Ornithine transcarbamylase deficiency. We said it causes orotic aciduria. صح؟ طيب. We said إنه إيش؟ The signs are hyperammonemia, decreased urea, and elevated glutamine. طيب. How do we know إذا الأوروتيك aciduria جانا من ornithine transcarbamylase ولا من ال UMP synthase? Simple. If we have increased ammonia levels, it's an OTC deficiency, ornithine transcarbamylase. Hello? Because we will have increased ammonia and you will see encephalopathy. But if the deficiency is in UMP synthase, there won't be ammonia. There will only be the megaloblastic anemia that can't be treated. Okay? طيب. We're finally done with the session. Few more practice questions about this lecture. نكون خلصنا. Megaloblastic anemia that does not respond. What could be the cause? We just said it, UMP synthase deficiency. Fluorouracil. What does it affect? We said thymidylate synthase. Major difference between purines and pyrimidines. I said it to you in the beginning, and the purine starts with the sugar PRPP بعدين البase, but pyrimidine starts with the base بعدين puts the PRPP. حتى the biggest piece of evidence I have is here. We made the orotic acid وبعدين the PRPP finally came with the UMP. عرفتوا؟ بس بالبيورين synthesis sorry تحملوني شوي. We start by doing the PRPP first, by making the sugar بعدين the bases وما عارف ايش. خلاص؟ But that's a good thing to keep in mind. طيب. 1-year-old boy is anemic, lethargic, elevated levels of uric acid, activity of uridine monophosphate is low. What will alleviate his symptoms? What should we give him? We said uridine. Regulated step in pyrimidine synthesis is what? Carbonyl phosphate synthetase 2. I already mentioned that. With that, البال session خلص. Wallah, it's been a long bal session. I'm not used to talking this much صراحة. Feel very dry. But I hope this was useful. Again, it might be a bit long, ممكن 3 hours, but guys, this is not a review. This is not a high-yield summary. This is simply everything you will ever need with post-midterm lectures in this PAL session. يعني والله العظيم guys, consider this as 13 different PAL sessions and I just happened to do them in one. يعني genuinely كأني I did 13 different PAL sessions for all of these lectures وحطيتهم in one video. It's everything you need. You guys are in good hands. Review this PAL session very well, review the slides with the graphs that I have. Inshallah, you will do amazing. Thank you guys so much. Feedback, please tell us where we can improve, what we did well. بإذن ta'ala, we will see improvements in the upcoming years. يعطيكم العافية. Best of luck.